Hey everybody, welcome to my live stream. Thanks for joining me. I'm really happy that you've come. So today's live stream is the last live stream before your Biology Paper 2 exam, which I imagine for most of you is tomorrow. So last big push to get this over with. So what we're going to be doing is going over any of your last minute questions, any topics you just need a little bit of a recap on. So at the moment, I've got quite a big list of requests from people that follow me on Instagram. So I'm going to start by working my way through those requests. And then I'm going to be asking you to give me some questions in the comments, and I'll work my way through those as well. I'll start by showing you the list of what everybody has requested at the moment. So you can see if that is useful for you. So that's the list of what I'm definitely going to go over. If you've got anything extra, just pop it in the comments and I'll get onto it as soon as I can. Okay, so top of the list is hormones and the menstrual cycle. So this is on both combined science and the biology only specification. And there's actually two levels to it. So there's stuff that foundation people need to know, and then there's extra stuff that higher tier people need to know. So we'll start with the foundation stuff and then we'll cover the higher tier stuff. Okay, so the menstrual cycle, it's a 28-day cycle and it takes place in the female body. And there are 4 main hormones that control it. So at foundation level, this is what you need to know. FSH is responsible for maturing the egg in the ovary. And so one egg will mature each month. Then LH is our second hormone, and it's responsible for ovulation, which is just when the egg moves out of the ovary and into the oviduct. And then the third and fourth hormone for foundation, they both do the same thing, and that is that they maintain the uterus lining. So the uterus lining is where the egg will implant if it gets fertilized. If it doesn't get fertilized, then the uterus lining will just break down and it'll leave the body via the vagina, and that's just known as the period. And that lasts 1 to 5 days, which is at the start of the cycle. So that's everything you need to know for foundation. So higher tier then, you have to know a little bit extra about each of these hormones. So you have to know their impact on each other. So we'll start again with FSH. It does the same thing in maturing the egg, but in addition to that, it stimulates estrogen production. Stimulates is a good word to use. It just means it causes it to happen. So estrogen is then stimulated to be produced, and it's produced by the ovaries. And then estrogen will start to build up the uterus lining. And then it does 2 other things that you need to know. First of all, it inhibits FSH, which means it stops any more FSH being made. This is really important because we don't want more than 1 egg maturing each month. If we did, we might risk having multiple births, say if there were like 5 eggs in the oviduct, and then that could equal 5 children, which is really dangerous for the mother and the baby. So estrogen inhibits FSH, but on the flip side, it stimulates LH production. And that just means it makes LH get produced. So LH, again, it just stimulates or causes ovulation on day 14. That's it for LH. There's not actually an extra thing to learn for it at higher tier level. But then progesterone, there's extra stuff to learn. Just like foundation, you need to know that it maintains the uterus lining, but then it inhibits both FSH and LH. So that's important to know. And it's produced by the empty follicle, which is where the egg left the ovary from. So that's where it comes from. Oh, and I forgot to say, FSH is produced in the pituitary gland, which is in the brain. So that is what travels from the brain all the way down to the female reproductive system. So my way of remembering that FSH is produced in the pituitary gland is FSH kind of sounds like fish. It's just missing the letter I. So it's like a little fish that has to swim through the bloodstream to get down to the female reproductive organs where it acts. Okay, so that's the recap for menstrual cycle at higher tier level. Let me know if that makes sense or if you've got anything extra you'd like to know in the comments. I'm going to hold up a little recap of that, and then you can take a screenshot if you would like these notes. So here you go. There's the menstrual cycle at higher tier level. I hope it's clear enough. I'm trying to hold it still. So take a screenshot if you want. I can also just stick it on my Instagram story after this. Okay, so that's the menstrual cycle covered, which was the first thing on my request list. I also had just hormones in general, and the second part of that was asking about adrenaline and thyroxine. So adrenaline and thyroxine, they are something that you need to know if you're studying higher tier. If you're studying foundation, you can just let this bit wash over you. So adrenaline, we'll start with that, is produced in the adrenal glands. And the adrenal glands are right above your kidneys. So if you can picture the kidneys, they're just on top of it. And adrenaline is also known as the stress hormone. So it's going to prepare your body for a stressful situation. And it's going to get it ready for either a fight or flight response. So for example, somebody jumps out at you and scares you, your adrenaline levels are going to whoosh, they're going to go up. So you need to know the changes that happen in the body when adrenaline is produced. And I'm sure you've felt them. They happen to everybody. So the first thing is that your breathing rate will increase. So you'll be breathing faster every minute. The second thing is that you're going to have your heart pumping faster. And then there's an impact of that. Two more things happen. Your brain and your muscles are supplied with more oxygen and more glucose. And if you remember from paper 1, what those two things combine to do in a certain process, it's respiration. So respiration makes energy, which is going to fuel you for that fight or flight response. So that's what adrenaline is for. Hey, Toby, thanks for joining us. Yeah, I do remember you. It's nice to see you virtually. Okay, so that is adrenaline. And then thyroxine, our second hormone for higher tier students, is produced in the thyroid gland, which is up here in your body. And it's responsible for controlling your basal metabolic rate. And your metabolic rate is just how quickly chemical reactions happen in the body. So when you're a child, you've got quite a high metabolic rate. When you're an adult, it sort of slows down. So it's important for growth and development. That's the big thing that it controls. You want to have your metabolic rate pretty steady in the body. You don't want it to be going up and down like crazy. So thyroxine is controlled by negative feedback. So again, that's a higher tier piece of content, but it's just basically that if thyroxine levels go up, the pituitary gland, which again is in the brain, it will detect that and it will release less of a hormone called TSH, which is thyroid stimulating hormone. If there's less TSH, it means the thyroid is stimulated less, so it makes less thyroxine. And so your levels come back down to normal. So it's just an example of negative feedback in the body. And the opposite would happen if the levels went too low. The pituitary gland would detect that and instead produce more TSH, and more thyroid stimulating hormone means that you would just have more thyroxine produced by the thyroid gland. Okay, so that's thyroxine covered. Next up, we have got the reflex arc. So that comes under the nervous system part of homeostasis and response. And it's basically what happens whenever something is triggered that you don't need the conscious part of your brain to be involved with. So that's the big difference between a nervous action and just any other nervous response. You just don't even need to think about it. You're not even aware that it's happening. I'm just getting a diagram up. I have got a little book of resources. So let's see if I can find that. Here we go. Yeah, and that's really important because it makes your response more rapid. So you're going to protect yourself from these horrible things like, I don't know, sticking your hand in the fire. That's going to probably mean you're going to have a reflex action. Okay, so a reflex action looks like this. I haven't actually I haven't written the names of each part. Why not try in the comments and see if you can say what each part is, and then we'll talk more about this part of it. So what does number 1, 2, 3, and 4, and 5 all stand for? What do you think? I know there is a bit of a lag. There always has been when I'm doing these live streams. I tried to set the latency the latency settings to be as fast as possible, but it is a bit slow. So, any ideas? What is each part of this? So we start with a stimulus. What do you call these type of cells that detect a stimulus? And then what is the name of each of these 3 neurons? See if you can get them in the right order. 2, 3, 4. And then 5, there's actually 2 pictures. These are examples of the name for 5. And then finally, that will produce a response. So what do we think those things are? We've got one answer. Yay! I'm not sure if this is very laggy or if maybe we're just taking a bit of time to get going. But well done, Eliza. You've got them spot on. So we've got our receptor cells. That's at the very start. They detect the stimulus. Then we've got our sensory neuron. In the middle is a relay neuron. My tip for remembering that is if you're doing a relay race, then you always are collecting a baton from somebody and passing it on to somebody else. So it's in the middle. And then we've got our motor neuron that's going to bring our electrical impulse all the way to number 5, which is the effectors. And there are 2 types of effectors you need to know. You can have muscles and you can have glands. So that's a muscle, and this is an example of a gland, which is the pancreas. And then the effectors actually carry out the response. So in the case of sticking your hand in the fire, the response would be that the muscles would contract, which means they like shorten, and that would pull your hand away from the fire. So that's just an important little exam tip. Always try and say what the response is. Don't just leave it there. Say what the response is. Is it going to be a muscle or is it going to be a gland releasing a hormone? What do you think? So try and spot it. What is the response? Hey, everybody that's just joined us. So we're working through some requests. We've got down to the Reflex Arc. If you've got any more questions, just pop them in the chat box and I'll be working through them as soon as I can. Okay, so while we're on the Reflex Arc, this is actually not technically just for reflex actions, but this bit at the bottom is just a recap of what a synapse is. So a synapse is the gap between the end of one neuron and the start of another. And what happens in the synapse is when electrical impulse reaches the end of the neuron, this neuron will release chemical messengers. You can call them neurotransmitters. You don't have to. You can just call them chemical messengers. And these then diffuse across the gap. Try and use that word diffuse. You want to get those those biological keywords in wherever you can. So it's diffusion because it's moving from high concentration over here to low concentration over here. And they then bind to receptors on the start of the next neuron. And when they bind, they cause another electrical impulse to be started in the second neuron. So synapses, they're actually really important. They do slow down the nervous response because diffusion is way slower than electrical impulse, but it makes sure that the message is going where it's meant to be going. If you just had all of your neurons directly touching each other, you would have messages getting sent across your entire brain or your body all at once. So it makes it a lot harder to coordinate a specific response. So that's why the body uses synapses. It's a good thing. Hey, everybody that's just joined. Like I said, just stick any questions in the comments. And thank you, Safia. I'm really glad you're finding it helpful. Okay, so the next topic is antibiotic resistant bacteria. Okay, so if you missed any of this, you can just go back in the video and watch that. Okay, so antibiotic resistance is when antibiotics, which are a type of drug that you take when you've got a bacterial infection, is when they don't work anymore. And it's really bad because it means that you're not going to be able to get your illness treated and you're probably going to have to try a different kind of drug instead. So it's a pretty bad thing because it's happening in hospitals. There are some superbugs happening. There's one you need to know. It's called MRSA. Those are all capital letters. It stands for methicillin-resistant Staphylococcus aureus. You don't need to know that, but the methicillin-resistant, that's just the name of the antibiotic and the fact that it's resistant to it. So MRSA is the one to learn. So you need to know how it happens and how we're trying to prevent it. So let me just check if I did a little graphic for this. I have got my book of resources, and that is in topic 1, or rather 5, of homeostasis and response. Or maybe it's not. It's in the next one. Let's have a quick look. Oh yeah, sorry, it's right at the end of inheritance and variation. Okay, so, I've got too many pages now. Here we go. Okay, yeah, so I have got a little diagram at the bottom of the ways that we prevent it. While I'm explaining what causes it, see if you can work out what each of these methods of prevention is. So what causes it is essentially a mutation, which is just a change in the DNA. That's happening all the time. Mutations happen continuously in our cells as well as bacteria cells. But most of the time, the DNA repairs them, or it actually has no impact on the phenotype, the physical characteristics of the organism. So we don't notice most mutations that happen. But very rarely, a mutation will come up that causes the bacteria to become resistant to the antibiotic, so it doesn't get killed by it. Now, when the antibiotic is used, all the other bacteria will die, except for those that have that mutation for the resistance. And if all the others are dead, only the mutated ones are alive, that means they're the only ones that are going to reproduce. And as we know, bacteria often reproduce asexually. So whenever they reproduce, that means that every single one of their offspring is a clone, and so they're all going to have the antibiotic resistance. So it's really bad news for you if you do get an antibiotic-resistant bacteria in your body. Okay, but we are trying to prevent this. It's really important that we keep antibiotics that work. So what are these 3 ways of preventing antibiotic resistance? I do see some answers. That is absolutely great. Okay, so when non-resistant bacteria are killed and resistant bacteria survives, is there less competition? Yes, Eliza, that's a question, not an answer. But absolutely spot on. Yeah, there's no competition because all the others have died, so they've got access to all the nutrients, all the oxygen, etc. So they survive and reproduce like crazy. Okay, great. Here's our first answer. So doctors should not prescribe antibiotics unless it's absolutely necessary. So that means if, for example, you go to your doctor and you've got a viral infection, they should not be giving you any antibiotics because they won't treat a viral infection. They also shouldn't give you antibiotics for anything that's non-serious. So if it's just like a common cough or something, they shouldn't give you antibiotics. You should just be left for your own immune system to help you get better. The second one, did anyone get that? Let's see in the comments. Yep, brilliant, Patty. You joined me for my last one, so you probably already knew this. You should finish your entire course of antibiotics. So this is just showing a little calendar with lots of ticks to show that they've finished every single day of their course. So that's a big one. You can do it, your family can do it, etc. Make sure you do it. Never stop early. If you do stop early because you start to feel better, then there might still be some bacteria in your body, and those might be some that could get a mutation for resistance. If you don't kill them while you can in the early stages, then that could happen, and then the infection can come right back again and you get ill again. And this time the antibiotic that you just had, it won't work anymore. Okay, number 3 then, I saw some good answers. Stop giving antibiotics to agricultural animals. So that's things like cattle, pigs, chickens, things like that. So anything for farming, you shouldn't be giving them antibiotics. This happens a lot in certain countries. In the UK, we're restricting the use of antibiotics in agriculture. So that's what we need to do. Thank you, Game Station. I'm afraid I can't do your test tomorrow. I've already done my GCSEs, so it's down to you, but you'll be great. Okay, so Anthony, you want to do inheritance? That is on the list, and it's next. So we're going to look at Punnett squares, which means that we need to understand all of the inheritance keywords. So let's take a look at that. All right, there are loads of keywords for inheritance. So I'm going to whiz through what they mean, and then maybe we'll try and do an example. Okay, let's see. Number 4. Okay. Well, for the notes I had prepared, I haven't actually done a definition of the words. I'll just explain them and then we'll try this Punnett square. Or maybe you can try it while I'm explaining if you already know. Work out what the offspring probabilities would be. Okay, so let's start off with the word allele. So an allele is just an alternative form of the same gene. So for example, you might have an allele for dimples, and you could either have the allele no dimples or the allele for dimples, or a combination of both. So in GCSE level, you're going to get your alleles given to you in pairs. So there'll be 2 alleles that could be the same, and if they are the same, they're called homozygous, or they could be different. And if they're different, they're called heterozygous. So my way to remember that is the prefix homo. And you've probably heard of homo in a few places. For example, homosexual means same sex. So homo just means same. So homozygous is 2 of the same alleles. So like a big D, for this example of dimples, it would be 2 big Ds or 2 little Ds. So if it is a big D, that means that it is dominant. And dominant alleles are always expressed, which means they're always shown even if there's a recessive allele. So it's like the stronger one. The dominant one is the strong one. So in this case, if you had this capital D, then you would have no dimples, because that's what it stands for. Now, you'll always get told what is dominant and what is recessive, with the exception of cystic fibrosis and polydactyly, because those are ones you have to learn. But we can go over that in a minute. Okay, and then last keywords. That might be it. Have I covered them all? Homozygous dominant, homozygous recessive, heterozygous. Yeah, that might be it. Hopefully I have not missed any. I think I saw somebody ask if I have a link. I do. Well, sort of. If you go onto my channel and you find my video called Inheritance Keywords, I will go through all of them for you with some nice little examples. So yeah, you can check that out there. Okay, so let's go through this one. We've got parents, and one of them is homozygous recessive, and one of them is heterozygous. So we would have to draw a Punnett square to work out the probability of their offspring having dimples or no dimples. So see if you can do that. You probably need a bit of paper for this if you're going to try it. So a homozygous recessive parent has a children with a heterozygous parent. See if you can do that. I'm going to write the answers and then we'll check what you got. So homozygous recessive means 2 little Ds because it's homozygous, so they're the same, and it's recessive, so it's the little Ds. And then heterozygous means we have 1 of each allele, or 2 different alleles. We're then going to work out our cross, and you just read down and across. So this is what it should look like. Here's our homozygous recessive parent, which is 2 little Ds, and then our heterozygous parent is 1 of each. And then we just cross them and we should get this answer. And then finally, we're going to just say what each combination stands for or what it would look like. I did miss a keyword. It's phenotype and genotype. So these are the genotypes, the 2 alleles together, and the phenotype is physically what they look like. I remember that because pH for phenotype can also be the pH for physical characteristic. So it's what you see, what the appearance is like. Okay, so the big D, little d means no dimples. because remember, the dominant allele is the one that takes control. And 2 little d's means that they have got dimples. And that's 50% of each, because there were 2 of each. If you had to write that as a probability, how would you write that? Don't worry, Laura, I know it's so much harder when you don't have paper. I wouldn't try it in my head. Definitely not in an exam. Always make sure you write it out. But of course you would. But anyway, 50%, 50%. How would you write that as a probability ratio? Any ideas? See if you can add in the comments, what would the ratio be for that? I'm not sure, maybe a bit laggy. Yep, well done, Paddy, that's spot on. 1 to 1. So when it's the same, it's 1 to 1. If it was 75 to 25, it would be 3 to 1. So that's how you do your probabilities. There's not very many options for probabilities when you do Punnett squares because it's only out of 4. So there you go. Okay, so inheritance, last thing then is you need to know sex inheritance. So sex is the chromosomes that you have got. You've got 23 pairs of chromosomes in every human cell of your body, but one of those determines your sex. So the female sex chromosomes are XX, and the male sex chromosomes are XY, like that. And then, ooh, it's very bright, sorry. You could be asked to work out or show that there's always a 50-50% chance of having a male or a female offspring. So you could be asked to draw a Punnett square and prove that. And all you do is exactly the same thing as over here. You take your female, you split them up. You take your male, you split them up. And then you carry out your cross and you say what the probabilities are. And again, it's going to work out exactly the same as last time. It'll be 50%. So I'll just do that quickly to prove it to you. But that's what you would need to do if you got a question saying prove that the inheritance of the sex character, the sex chromosomes is always 50%, and you would have to draw a Punnett square. Okay. So again, if you do want any of this information, you can take a little screenshot now. Or if you follow me on Instagram, I will pop these up in my stories later. There you go. That's your chance for a screenshot. Okay, so let's see what's up next. We have got all the way down to Punnett squares. If you missed anything from up here, you can go back in the video and catch up on that. Okay, so next up is kidneys. Now, the next bits on my request list are biology only, i.e. triple. So very quickly, if there is anybody studying combined science that urgently wants something covered, let me know and I'll do it first because it applies to everybody. But if not, then we'll go straight on to these biology only topics. Apart from required practicals, now that is obviously on both sides of the course. Okay, Oriana, yeah, you want some required practicals. Game Station, I'm afraid I haven't got any of the biology only videos created yet. I just didn't have time. I only started my videos like 2 months ago, a little bit more than that now. So I just didn't have time, sorry. But I can answer your questions here. Okay, so Patty, you've asked for the decay practical. That's the biology only one. And I think everybody else seems happy. So fertility. Okay, we can go over that. Do you mean, for example, infertility? And you want to go over the infertility treatments? Would that be right? Maybe? Yeah, we'll go over that really quickly. Yep. All right. And then Fatima, yep, we'll go over controlling body temperature. That's biology only, so we'll do that after. Okay, so infertility treatments and IVF, yep. So there are lots of reasons why a person could be infertile. If it's a woman, she could have blocked or damaged oviducts. She might not have the right levels of those reproductive hormones that we discussed earlier. And so she's not maturing or releasing her eggs, for example. In males, it could be that there's a defect with the sperm. For example, the tail is damaged, so it can't swim properly to get to the egg. could be a low sperm count, so there's just not enough getting released. Or sometimes doctors just can't find a reason for infertility. So they can try and treat it. With a woman, one of the first things that they could try is to give her the reproductive hormones, FSH and LH. So those are combined, called fertility drugs, because FSH matures the eggs and LH causes ovulation. So if you give those to a woman, it might increase her chances of becoming fertile and having a baby. So if that doesn't work, they could offer the woman IVF, which is standing for in vitro fertilization. And that basically means that the fertilization is going to happen in the lab. So there are 5 main stages to IVF. So the first stage is what I just mentioned. This is the woman and she's receiving the fertility drugs, FSH and LH. See if you can tell me what's happening in steps 2 and 3. I'm going to have a go at labeling this while you do that. But see if you can tell me what's step 2 and what is step 3. I just saw a little request for going over the ratios. Yeah, I can do that in a second. Yeah, I will show you again. Sorry, it's quite brief. I just want to get the answers down, as I know some people just like seeing the answers. Okay, so step 1 was FSH and LH being given to the woman. So FSH makes the eggs mature and LH causes the ovulation. Then once those eggs are mature, you're going to extract them. So that can be quite a painful process. You do need to know some of the issues of IVF, so that's one of them. It's pretty physically stressful and emotionally stressful. Okay, so the eggs get extracted and then they're going to get fertilized with a sperm donation in the lab, often the woman's partner. So that happens in a lab. The scientists in the lab will monitor that to make sure that fertilization is happening. And then over here, you can see there's a microscope. That's important because microscopy comes up across the specification. You need to know that its developments in microscopy have allowed us to do this. It's only because we've got really good microscopes that we can carry out IVF now. So they will monitor the fertilized egg to make sure that it develops into an early embryo. And over here, you can see we've got 2 early embryos that have developed correctly, and they are getting inserted into the mother's uterus. So remember, that's where the fertilized egg or early embryos will implant into the uterus lining. So that's the last step. Okay, so I'll just fill those in. And then if you want to, you can take a screenshot or you can just take a look. So monitor to see if the early embryos are developing properly. That's really important because they don't always. That could be one reason why a woman is also infertile, if the embryo doesn't develop properly and then won't implant. Can you tell me in the comments, why are only 1 or 2 embryos inserted? Why not insert like 4 or 5 to increase your chance of success? Why would we not do that? Let me know while I finish up writing. Okay, and can you both, Patty and Eliza, well done. Can you all tell me, please, why we don't want multiple births? So here's our last steps. If you want to take a screenshot, now is your chance. Yeah, very good, Oriana. If too many babies came to term, it could be risky. And that's for both the mothers and the babies. If the babies are born with a low birth weight, that means they're more likely to have problems in their early life. Very good. Dangerous to mother and child. That's spot on. Okay, so the issues of IVF are it's extremely emotionally and physically stressful, and there's really high levels of non-success. Have I phrased that right? Let's phrase it the other way. There are low levels of success. So even though you go through all of this hard work and time and money, it's expensive, it might not even work. And it is expensive, so it's a drain on the NHS resources. Okay, so that's infertility treatments. Okay, shall we go over some required practicals? I think that could be something good because that applies to everybody. So I haven't done lots of notes on these, but let's just check. Okay, so the first one in combined science are reaction times. So that's for everybody. You all need to know that. That's quite a straightforward one. Essentially, you're going to test the effect of a factor on reaction time. It could be lots of different factors. There's no one factor that you need to know. So it could be caffeine. You could have a cup of coffee or a caffeinated drink, and then test reaction time before that, and then after having consumed the caffeine. It could be lack of sleep. It could be another different type of drug like alcohol. It could be anything really. Age. Yeah, there's loads of different ways you could test reaction time. Then the method of how you test it tends to be the ruler drop test. That's the easiest one to describe. You can describe other methods, perhaps some better methods that are going to involve digital recording of your reaction time. But the ruler drop test one is a classic. So you would have your ruler. You need to describe in your method really clearly where the ruler is placed. So it needs to be placed right above the thumb and forefinger at point 0. So you need to have it at point 0. The ruler is dropped without any indication or warning. So it's not like you're going to say 3, 2, 1, drop, because you're going to catch it a lot faster than you would. It's not testing your reaction times. So it's going to be dropped without warning. It's caught. And then where it is caught, that number is recorded, say 15 centimeters, 40 centimeters, whatever. And then you need to use a conversion table. Don't forget that step. Giving you 15 centimeters, that's not a time, that's a length, that's in centimeters. So you need to convert it to a reaction time using a conversion table. And you don't need to have one or explain what that is. You just need to say that you use it to work out the reaction time. Then you're going to repeat it, taking on whatever factor it is that you're testing, and you're going to repeat it multiple times and average your results and get some results. So Paddy's made a good point. If you do use something digital like a computer to record your reaction time, that's a lot more precise because it's going to give you your answer to many decimal points. That's what precision is. And so that's going to give you a better indication of your reaction time. And yeah, no indication. So even though I said you're not going to count down 3, 2, 1, you could still be giving your partner subconscious cues that you're going to be dropping something or when you're going to drop it. So you might you might give it away without intentionally trying to give it away. So it removes human error and bias, which is important. Thank you, Safia, for answering Anissa's question. I have already covered antibiotic resistance, so you can flick back a bit to see that. Okay, so that's our first required practical for combined science. So on the homeostasis and response topic, there's also a required practical for biology-only students, and that is the effect of light or gravity on the growth of seedlings. So this is going to test your understanding of the hormone auxin and its response to light and gravity, so phototrophism and gravitrophism. I didn't see anybody asking for that practical. Do you want me to go over that one? Let me know in the comments. I'll just talk you through it if you do. If you don't, I'll move on to the next one. Okay, got a lot of people saying yes. Sorry to the people that said no. We'll move on after anyway. So you're going to get given one or the other, light or gravity. So what you need to do is set up your seedlings. If for gravity, you'll just put them on their sides, and then you're gonna monitor them. You need to make sure that the conditions are constant. So you need to make sure that the soil has got the right levels of nutrient, moisture, water, etc. And then you're gonna most likely with this practical have to explain it. because there's quite a lot in the specification about the results. So yeah, I can do plant hormones as well. So I'll kind of talk about the plant hormones first, and then we'll make sense of it with this practical. So auxin causes unequal cell growth, and that's because it normally gets unequally distributed in a shoot. So I've got a nice diagram somewhere. Let me try and find it. Here we go. So if we look at phototropism first, you can see that auxin moves to the shaded side of the shoot. So this is a shoot that's growing. Now, you don't actually have to learn that auxin causes elongation in shoots and inhibits elongation in roots. You can just say that it causes unequal growth rates in the cells. You don't have to learn it, but if you have learned it, you absolutely can say that this shaded side would grow longer, and that then causes the shoot to grow towards the light. So that's positive phototrophism. In gravitrophism, the shoot will want to grow away from the force of gravity, and the root will want to grow towards the force of gravity. So auxin actually has a different response in the shoots than in the roots. In both cases, the auxin will move towards the force of gravity. So it will drop to the bottom part of the plant. So in the shoot, you can see it here at the bottom, and in the root, it's here at the bottom. Because there's more auxin here at the bottom in the shoot, that again causes elongation, or you can say the unequal growth rate in the cells. And so it will start to curve up away from gravity. But in the roots, it actually does the opposite. It inhibits cell elongation. So it's actually going to slow down the growth of cells on this side. So the top side is growing faster, and that sort of makes it curve down towards the force of gravity. Okay, so that is how auxin works. So in your required practical, you'd have to set up conditions that show this. So for gravitrophism, if you're doing it, for example, in shoots, you need to make sure that light isn't affecting it. So you would really, that's a big control factor or control variable. You would need to keep the light constant. We've got a little tip here for a way to remember it. It's stim in the shoots and restricts in the roots. Yeah, I like that, Megan. That's really nice. Don't worry, though, if you haven't learned that, you don't need to for AQA. AQA explicitly say that you could just learn that it causes unequal growth rates in the cells. I've seen another question there. Any tips for answering 4 to 6 markers? Or, yeah, tips. Think about your points. Make sure you've got 4 discrete or separate, really clear points. Don't try and waffle. You're not going to get extra points just by waffling on. You need to get your keywords in. You can bullet point them if you want to make sure that you've got 4 exactly, and use the information given. Often in these long mark questions, you're going to be given something to talk about. It might only be worth 1 or 2 of the marks out of that 4 or 6 mark question, but if they've given you information, there's a reason for it, and you'll have to use it somewhere in your answer. So just try and spot what angle the examiners are poking at. And yeah, put down more points rather than less if you've got a few extra points. There's no negative marking in biology. So if you've got the time and you're not sure completely what points to put down, you can always put down more, and they'll just mark the ones that are right. Okay, so I think we've covered roughly the required practical. Has anybody got any other questions or any other tips for each other? That's really nice to see you sharing that. Okay, so then the last required practicals, there are no required practicals in the inheritance topic, which is great. But in the last topic, we've got 2 more. So in ecology, we've got the transect one and quadrat one, which is measuring the population of a species and also seeing how its distribution changes with the effect of another factor. So I'll go over that quickly. So that comes up also in the specification under sampling. So it's in my opinion, is probably quite likely to come up. They made it come up a lot in the old specification, which also had this content on it. So it could continue to come up again. So here is a transx. These are used randomly to work out the population in a large area, like a school field. You've probably all used them. So you would use it randomly in a couple of ways. The first way is not so good. It's when you're blindfolded and you walk around and then you throw the quadrats, but that's not the best method. The better method is to divide your sample area into a grid, then assign each square in the grid a number, and then use a random number generator to choose the squares that you're going to sample, and that's where you place your quadrat. So that's the best way to do it. Okay, then you will take as many samples as possible. That's going to make your results more valid, which means they're probably going to show you the correct distribution of the species. And then you take an average. It's really likely you could get asked about the mean, the median, the mode, even the range within this. There's a lot of maths in this topic, so just check your maths is up to scratch. I have got a video on maths and biology. It's quite long. I think it's like an hour and a little bit. But if you have time and you're confident in all your other content, you can check that out. Check, how to do standard form and things like that. Okay, and then there are transx. So transx are like this. You are going to have a measuring tape and you place your quadrat at equal intervals along your transx. So for example, if this were a 50-meter transx, you would maybe place them every 5 meters. So you'd get 10 results. So transx are used when you want to see the change in distribution as another factor changes. So for example, if you had, let's say, a woodland at one end and the transect was going in there, there's going to be less light, perhaps there's more moisture. So you're going to pick one of those factors and you're going to see how the distribution changes. So what you also need to do is measure the light, for example, at each quadrat interval as well. And so you can record as light changes, the distribution of daisies also changes like this, and you work out that relationship. So you want to repeat your transect as well. So you'll have one line here, maybe you'll go 5 meters this way and do another one, 5 meters this way and do another one. So always we want to try and repeat our results when possible. Okay, so that's how you use your transects. The last thing is something I mentioned in my ecology live stream. It's kind of something I noticed and was a bit surprised about, I guess. It comes up in the maths part of the specification that you need to understand the principles of sampling. And we've just covered those two, but there's like an extra one that's mentioned in the AQA textbook. So I'm just going to mention it just in the off chance it comes up. And that's the mark, release, recapture methods. So this is used for often more motile organisms, which just means ones that can move. So for example, snails or like ladybirds or, I don't know, little bugs. And essentially what you do is you're going to go out and sample them on 2 days at a different time, like a week apart or a few days apart. The first time you capture your organisms and you're going to mark them. So you're going to put a little blob of paint or something like that on them. And you record how many you captured. So maybe you recorded 40 the first time. You release them, that's the release part. And then you're going to go out, let's say a week later, and you're going to capture this organism again. And you're going to count how many you capture. And then let's say you caught 20 and only 5 of the 20 had the paint that you put on them before. This is then going to be used in an equation to work out the population size. So here's the equation. I think that they'll give it to you because it's not explicitly in the specification that you have to memorize this. So the estimated population size is your first sample, which I said would be 40, multiplied by your second sample, which was 20. You didn't find as many the second time. And then you divide it by the number of recaptured marked individuals, which would be 5. So you just pop those numbers in and you get your answer, and it was 160. So that's our mark, release, recapture method. It could come up. It's kind of sneakily put in the math part of the specification. But there you go. That's a little recap of it. I'll hold this here. If anybody wants to take a screenshot, now is your chance. There's a question there that I haven't gone over because the answer is already there. But you can take a look at that yourself. Okay, we've got some questions, or comments at least. Can we bullet point longer answer questions because they've removed the using good English? Yes, you absolutely can. Bullet points are great. You should use them if they help you. And Ray wrong, here you go. And Patty, I do think that the formula will be given to you. I think there would be a lot of complaints if it wasn't, although don't quote me on that. So I think probably, but yeah, I don't know. We'll see. Okay, I'll go through the very last required practical, which is on the biology only part of the specification for ecology, and that is the decay practical. So for this practical, you need to know about milk and how its pH changes as it decays. So milk contains the sugar lactose, and bacteria will break that sugar down into lactic acids. Remember, bacteria are what carry out decomposition along with some fungi. So as milk decays, the pH is going to get lower because lactic acid, it's an acid, so it's acidic. So pH is going to drop. So what you're going to do is you're going to get your fresh milk and you're going to leave it for a few days because decay will take some time. So to be able to see how the pH changes, you obviously need to use an indicator. An indicator you should use is universal indicator. It's going to be really easy to see those color changes. So you'll use your indicator chart alongside it to record what's happening to the pH. And you should do that every 12 or 24 hours every day. You should check how the pH is changing. So the big factor that you're going to change, your independent variable, is the temperature. So you're going to want to have different temperatures. You might put one in a fridge, you might put one at room temperature, and you might put one beaker of the milk at, let's say, like 30 degrees, maybe in an incubator somewhere. You add your universal indicator and you record the pH every day. And that's it, really. You're going to obviously see that the ones that are in warmer conditions are going to decay faster because warmth is a condition for decay. And if you're doing triple science, you'll have learned about that. Grace Mills, can you use phenolphthalein? The AQA specification, I'm pretty sure, says universal indicator. I'm not sure is the honest answer. We've got somebody saying yes, so I think you probably could. It's been a long time since I did my chemistry A-level, so I can't even remember exactly how the color changes for that. But maybe, I don't know. If you're unsure, go for universal indicator because I've seen that. And you use creosote red, Ellie. Yeah, I think Patty, yeah. As long as you say a method of recording the pH, like a pH probe, then that's fine. You couldn't get marked down for that. Yeah. And actually pH probes, they're good. They're really precise. Again, they're going to be more accurate than just using color because color is always a little bit subjective or open to human error as we interpret them slightly different. Okay. So that's our required practicals done. Yay. Okay, so that is everything covered down to here, apart from kidneys, because that's our biology only stuff. I'm afraid I didn't quite write down everything that anybody else has said. So if there is anything else you'd like me to cover from combined science before I get on to biology only stuff, I'll put little stars next to that because it's not very obvious. Then let me know. So is there anything else you want covered from combined science? Ryan, I'm afraid I've already covered the menstrual cycle. I did it right at the start of the live stream. So if you just flick back and go to very start, then you can revise that. Patty, yes, I'll go over that. That's biology only. as well. I've got it under understanding genetics. Cycles, Ray wrong. So you mean like the water, decay, and carbon cycles? Yeah, sure. Okay, and then we're going to do classification, and then we'll finally move on and do kidneys. So here we go. Here's our cycles. We'll do this really fast. I recommend just taking a screenshot so you can look at it later. So over here, we've got the water cycle. Here are the three parts of it. We first of all got evaporation. We're going from a puddle into the water vapor in the air. It will then eventually condense, which is where it turns back into a liquid from the water vapor. And eventually, when it reaches that critical mass, it will precipitate, and that means it falls as either rain, sleet, hail, snow, etc. So that's the water cycle, short and sweet. Yep, the nitrogen cycle is biology only, and actually you don't really need to know like a whole cycle, it's just a bit of it. Decay, you need to know the two types of decomposers. They are bacteria and fungi. And decay is really important because it's going to allow nutrients, mineral ions, to be recycled. So when they break down the dead organism, they're going to release the mineral ions from that dead organism. They're also going to release carbon dioxide into the atmosphere because these are both respiring as they carry out the decay process. And it actually comes out as part of the digestion of the dead organism. So include that in your answer as well. The digestion or feeding on the dead organism releases CO2. Okay, and then finally, we've got the carbon cycle, which is a slightly longer cycle. And you can see it incorporates a little bit of the decay cycle. So we've got death and then decay. So starting at number one, we've got feeding. Here we've got producer, a plant, and a primary consumer, a little bunny, and it's feeding. It then dies. Both of them are capable of dying. So there's death, decay, which is over here, we just discussed. And then an alternative to decay is fossilization. So that's whenever the dead organisms get turned into fossil fuels, coal, oil, and natural gas. So that takes millions of years, as you probably know from chemistry, and is only under the correct conditions. Over here then, we've got the burning of the fossil fuels in this factory, and that is called combustion. So that's when they're burned, normally in the presence of oxygen, and that will then cause carbon dioxide to be released into the atmosphere. Okay, in addition to that, respiration will also cause carbon dioxide to be released. All living things carry out respiration. So we've got arrows coming from the plants and the bunny, the animal, and that releases carbon dioxide into the air. And then we've got a different colored arrow for number 7, which is photosynthesis. So that's the only process in the carbon cycle here that will remove the carbon dioxide from the atmosphere. So it's really, really vital that we've got lots of plants on our planet to carry out that process to reduce carbon dioxide. That's why deforestation is such a big problem. But that comes up a bit more in the later part of this topic. And just to point out, I have got CO2, this is in the ocean, just to highlight that it can also dissolve into water. So there you go. That's all of our cycles for you, Ray Wrong. Okay, Anissa, you wanted classification. I've got a little mnemonic that's going to help you remember that. It's definitely keep ponds clean or frogs get sick. So that's going to give you the letter for each part of the classification hierarchy. Linnaeus came up with all of them apart from the D. Do you know what they stand for? Well, I'm going to show you just to keep things quick for those of you that need to shoot off. So here are our letters. So I'll tell you my mnemonic again. Maybe it'll help you. You might remember it in the exam. So definitely keep ponds clean or frogs get sick. So that helps you remember the order. And then, of course, you need to remember the actual scientific names, which are domain, kingdom, phylum, class, order, family, genus, species. The bottom two make up the binomial name. which is the two-part name of a species. So, for example, we are Homo sapiens. Homo sapiens. I didn't say that right. So, Homo sapiens is just humans. Okay, you need to know a little bit more about the top level of classification, which is domain. So, Woos, I'm not sure if I ever say his name right. Woos came up with this. I'm going to write his name down so you can see how it's spelled. He came up with this in the 1980s, and he came up with it due to improvements in microscopes. So this kind of, somebody on Instagram messaged me asking about a 4-mark question, and it was about how classification systems have changed. So you could get a question like this, and you need to explain that improvements in microscopes have allowed us to see the internal structures of cells. So back in Linnaeus's time, they could not really observe that. They could only see organelles and large structures. But now we've got electron microscopes, so we can see the internal structures of these organelles. So that's a huge advancement. In addition, we've progressed our understanding of biochemistry. So we can understand the biochemistry of a cell, which are those reactions that are taking place inside. So that's another huge improvement and has led to different models and this new level of classification. Okay, so domains, there are 3 that you need to know. We've got archaea, bacteria, and eukaryota, or BAE, if you remember it like that, B-A-E. Domain is BAE. There you go. So you need to know what they are. The archaea are the primitive bacteria. Bacteria are like true bacteria. Prokaryotes can fall under both of those, just as a side note. And then eukaryotes are basically everything else, like plants, animals, fungi, and protists. Those are all eukaryotes. Okay, so that is classification. I think that's everything you need to know for it, hopefully. Okay, let's check just if we have any other questions. Could we cover the transfer of biomass and energy in the food chain? And no, I haven't covered the brain and the eye, but I do have some nice drawings for that. So yeah, I can do that as well. Okay, so transfer of biomass and energy in the food chains. I think that comes up in biology only part of the spec. Yes, it does. Okay, so biomass is obviously transferred through eating other organisms. Not all of it is transferred. Well, the first big area of loss is that not all of an organism is eaten. The hard parts, like the bone and cartilage, tends to be discarded. And same for plants, even when a tree is eaten, the bark doesn't tend to be eaten. It will be the leaves or the fruits of the tree. So not all of the organism is eaten. Okay, then there are other ways that biomass is lost as you go up the levels, the trophic levels, from bottom to top. So feces, animals and us, we all go to the bathroom and we lose biomass every time we do that. So that's the first one. Waste, we lose other products when we respire. Carbon dioxide and water come out in our breath. We lose a lot of water in our urine and urea as well. We can go over that when we do the kidneys. And obviously we are moving organisms. Animals run around, walk around, or just if you're a sloth, sort of breathe a bit. And as we move, we are going to use up lots of energy through respiration. For animals like us that maintain a constant body temperature, a lot of that is also used, a lot of energy and biomass is also used maintaining that constant body temperature. So if you think about farming techniques to try and reduce the loss of biomass, farmers will limit movement and will control the temperature surrounding their farm animals. And basically they'll keep it nice and warm so that the animals don't have to use up any of their own energy to maintain their body temperature. Okay, so that is that's how energy is lost in food chains. Okay, so Patty, you wanted to go over some of the big names in understanding of genetics. So let's do that now. Here we go. So here's a little bit of, oh, Laura, sorry, just before we do move on, you've got a question about what we just did. How do you do the scale at the bottom of the pyramid? Okay, so this is just a scale like you would do for any graph. You get your biggest number, which is always going to be your producer in almost every case. And you need to divide it in two and then make a scale that fits there. So you divide it in two because your pyramids need to stack on top of each other equally. So I always suggest, before you do anything else, is you just get your ruler and pencil, and whatever graph bit of paper you've been given in the exam, you just draw a line straight down the middle of it so that you know where your center point is. And then let's say, for example, you had a number of 800 for the producer. You're just going to have 400 either side of your pencil line in the middle. And then you make your scale from that. So just like you make any scale for a graph, you need to make sure that each square is worth the same amount every time. And then for each level above it, you're going to then use that scale and try and make sure your scale is stretched out and uses all of the graph paper if possible. And then you just do the same thing for every level above. You just divide the number in two, and half goes on either side of your middle pencil line. Okay, so the main names that you need to know, these are the ones for biology only. In regards to understanding genetics, you also need to know about a little bit more about Darwin and Wallace. So you need to know about, actually, there's only really one main name here, I guess. It's Mendel. So we'll recap this. Somebody else had asked for details on this. So Mendel, he carried out those breeding experiments in plants, and he basically found out that there definitely was a mechanism of inheritance, and he called that units. So he knew something was being passed on, but he had no idea what, but he worked it out because he did loads of experiments with like pea plants. Okay, so he worked it out and he knew that that definitely was happening, but it wasn't until the 19th century that we actually got good enough microscopes to record the behavior of chromosomes during cell division. So actually seeing that they sort of line up across the middle, and then they move to the poles, and then the cell splits in two. So we learned that chromosomes are moving around and getting split into daughter cells. And then in the early 20th century, the microscopes got a bit better again, and we saw that chromosomes are behaving in the same way as units, so that they are getting passed on to the offspring. And we actually then worked out that the units are genes. So the genes are on the chromosomes, and they are what cause you to inherit characteristics. And then finally, can't really read that, in the mid-20th century, 1928, you don't need to know that date, but it's good to know if you like biology, the structure of DNA was finally discovered. So we knew a little bit more about that double helix, and then we found out the mechanism of how genes actually work. So like we just learned about dominant, recessive, etc. Yeah, gene function is figured out. Okay, so those are the main names for understanding genetics. Right, let's do kidneys. Sorry, it's taken a long time to get there, but kidneys is next for biology only people. Right, kidneys. There's a good chance that if you're in a top set and you've got a teacher that teaches A-level, that they might have taught you more than you need to know, because it does come up in the A-level specification. But we're going to keep it nice and simple. We're going to keep it at GCSE level. So this is what you need to know. So the kidneys are going to be really important for filtering out urea. So let's talk about how urea comes to be in the body. So first of all, you will have some protein in your diet, whether that's through meats or whether it's through things like chickpeas or nuts, etc. And when you digest the proteins, sometimes you might have too much. You can also get excess protein in your body if you've got like worn out tissues that your body wants to recycle, because nothing lasts forever in your body really. So those excess proteins they will be broken down into amino acids. You'll have learned that from your digestion topic in paper 1. And then those excess amino acids will travel to the liver to get processed. So what the liver does is it carries out deamination. And deamination is whenever the amino acids are deaminated and they're converted into ammonia. Now, ammonia is toxic. If you've ever used it in chemistry, it will have a nice little hazard symbol because it's pretty dangerous. So the body immediately converts the toxic ammonia into urea. And I've given it this little purple dot symbol. So it goes from amino acids to the toxic ammonia and then to urea. So that's sort of 3 things to learn there. Then we're going to deal with the urea because the body still doesn't have any use for it. And if too much of it builds up, then it's also a little bit dangerous in the body too. So the urea, it'll travel in the bloodstream from the liver all the way down to the kidneys. And the kidneys are like a big filter, a big sieve. So they will filter out all of these things. So they'll filter out glucose, urea, water, and mineral ions. That's just a little salt to try and show you some mineral ions. So the kidneys filter out all of this from the blood. But then it completely reabsorbs all of the glucose, which is why I've done it in pink, because it's the only one that immediately gets fully reabsorbed. Okay, so the rest of them don't get reabsorbed unless they're needed. The urea never gets reabsorbed. It's a complete waste product. The body just wants to get rid of it. So it will get chucked out in your urine. But if your body is low on water or low on mineral salts, then those will get reabsorbed back into the blood. So it will be monitoring and making sure that you've got enough of both of those. Okay, so the way that happens is through ADH, at least for the water. So if you're doing higher tier, this bit in the bubble is higher tier only. So Raj, yes, I have made all my videos and things just using the AQA spec. So I'm afraid this is AQA specific, but a lot of it overlaps with Edexcel, so it might still help you. Okay, so ADH is secreted by the pituitary gland, which is up, probably too high. It's up in your brain, and it's only released if your blood concentration is too concentrated, i.e. you've not got enough water in your blood, so your blood's really concentrated. And ADH will travel from your pituitary gland all the way to the kidneys, and in your kidneys you've got millions of little tubules, which are just like it sounds, tiny tubes. And it causes the tubules to become more permeable. And permeable just means that things can move through them more easily. So specifically, water is going to get reabsorbed back into the blood because the tubules are more permeable. So that's one way of bringing your water levels in the body back up. So you're going to reabsorb more water. Okay, so this ADH is an example of negative feedback. So the pituitary gland is monitoring the amount of water in the blood, and let's say there's too much, it is going to stop producing ADH so that your kidneys, they don't reabsorb the water. So remember, negative feedback is just making sure that things stay at the set points of the perfect optimal body conditions. So that's one example of negative feedback. Yeah, that's spot on, Megan. ADH increases permeability of kidney tubules. Very good. Any other questions? How would we interpret tables and bar charts? I'm looking through the AQA spec and this just cropped up. I mean, it completely depends on what the question relates to, Megan. I always just suggest make sure you look really carefully at the numbers, see if you can spot patterns. If, for example, it's a really simple thing like the bar increases by twice as much, put that phrase in there, twice as much, 3 times as much, 4 times as much. Do some number crunching on your calculator. Don't just say it increases. Try and put something extra in there for your answer. Okay, so that is the kidney. The other last little bit is that dialysis can be used if the kidney stops working. And this is just a way of using artificial filtration. So your kidneys aren't working, you're going to use a dialysis machine. The dialysis machine will have dialysis fluid, and it's going to have the person's blood pass through the machine and separated with a partially permeable membrane. And so it's going to allow the urea to diffuse across the membrane, out of the blood, because we're cleaning the blood, and into the dialysis fluid, where it's going to be taken away. The blood and the dialysis fluid move in opposite directions to each other. The dialysis fluid will have the same level of glucose and mineral ions as the blood, like the right level, so that the person doesn't lose any glucose, because obviously glucose would move from high to low if there wasn't any in the dialysis fluid. So you need to make sure that that stays equal on both sides. And that's it, really. That's it for dialysis and the kidneys. Okie dokie. So that's kidneys crossed off on our list. Okay, I'm just going to put ticks, and I'm going to hold it up so that people who have joined us more recently can see what's already been covered. And so if there are any parts that you missed, you can go back a little bit in the live stream and find those. So Coral, I'm afraid we've already done the required practicals, so take a look at that. Okay, so next up are parts of the eye. I'll just answer these comments while I'm searching for it. So Siraj, any advice? I feel like I know nothing due to not being able to revise a lot. What would you do if I was in your position? So I would 100% get on the AQA website and download their specification, read through it and see which bits you already know, and just leave those aside. Focus on the bits that you haven't yet got in your head, and spend the rest of the evening just trying to memorize them. So make maybe little headings on some paper, hide the specification or close your laptop if that's what you're using, and see what you can write down from memory, and then open it back up and see what you missed and add that on in a different color. If there's a part of the specification you don't understand, watch my paper 2 video. That's my big video. It's 1 hour, 42 minutes. So you have got time to watch all of it if you want. And it covers all of the content for paper 2. So you've got time to do that. But yeah, get the specification and make sure you focus on the bits that you're not confident with yet. And you'll be fine. You probably know more than you think. Okay, so parts of the eye. That is biology only content. I really like this part because the eye is pretty cool. If you got to do a dissection of it, that's cool as well. I'm going to just take a wee sip of water. I'm a wee bit parched. Okay, I should put the lid back on. I don't want to spill up my laptop. Oh, thank you for anybody that has watched it. That's really nice. Thank you for the feedback. And Laura, the double speed. Yeah, I haven't tried that. I'm sure I sound pretty funny on double speed. Sometimes I talk a bit fast anyway, so I don't know. Okay, here's the eye. So you need to know the parts of the eye. If you're not sure, just get a diagram and practice covering it up and labeling them. You also need to know what each part does. So I'll just go around and talk about that first. So the cornea is the front part of the sclera. The sclera is just the protective layer that goes all around the eye, and it protects it. But the cornea is transparent, which of course is important because we need to let light in. It's coming this way, by the way. That's the front of the eye. And it's also going to be the first point of the eye that refracts the light. So when the light comes in, it needs to be focused on the back of the eye, which is the retina. So we need to refract it. So from physics, hopefully you know that means light changes direction. So it will come in, and then the iris is the colored part of your eye. Mine are sort of greeny, browny, gray, whatever, I don't know. So your iris is responsible for protecting the eye. If it's really bright light, your iris will really expand, so you've only got a little tiny pupil, so you don't let too much light in or damage your light sensitive cells on the retina. If it's pretty dim light, the iris will open to let more light in so that you get enough light to actually see. Okay, the pupil, I haven't labeled it, but it's just this empty space here. That's the black part of your eye where the light actually enters. Oh, I forgot my label here. This is the lens. You can see it looks like a sort of sac sitting in the middle of the eye, and it is supported by two things. The suspensory ligaments. You can't really see those. I used green, not the best color. But anyway, they are either side of the lens, and they are attached to the ciliary muscles, which again are on both sides. So the ciliary ligaments, the ciliary muscles, they can contract and relax, and the suspensory ligaments, they can be loose or they can be taut, which means they're like all stretched out. Okay, and they're going to be both working together to control the thickness of the lens, and we'll talk about that over here. The lens is then your second part that is going to refract the light, and it's responsible for the fine focusing of the light. So making sure that the light focuses, which means comes together perfectly in the right place on the retina. So not in front of it, not behind it, exactly on the retina cells. And that is called accommodation. So accommodation is focusing of the light on the retina by changing the shape of the lens. Okay, and then finally, I've kind of said it, the retina has your light-sensitive cell, and then the optic nerve is this little area here. I've just drawn a neuron. It's kind of wrong. Nerve is loads of neurons. But anyway, the optic nerve is what takes the message of what you're seeing to the brain to actually be processed and turned into an image. Okay, so here's what happens when you want to look at a near object. So here's my way, here's my way of remembering it. If you were looking at a contract, let's say you just got a new job and you had a contract and you were looking at it, then you would have your ciliary muscles contracting. It's kind of the same word. So if you're looking at wrong side. If you're looking at a contract, the ciliary muscles are contracting. They are squeezing tight together. The suspensory ligaments kind of do the opposite. They loosen, so they go slack is another word you can use. And the result of that is that the lens becomes thicker. If you're looking at something really close, that means the light's coming in quite spread out. So you need to refract it or bend it quite strongly to focus on the retina. So you need a nice thick lens for more refraction. I suggest taking a little screenshot of that. Here's your chance now. And so you can take another look at that. I haven't written down what happens with the far object or the distant object, but it is just the opposite. So the ciliary muscles will relax. That causes the suspensory ligaments to become taut, which means they're pulled tight. And then finally, the lens will become thinner, so there is less refraction. So distant objects are literally just the opposite of that. Okay. So then there's a second part of the eye. Whew, it's quite a big topic. And this is the defects of the eye. So we've got myopia and hyperopia. So we'll look at each of those defects in turn. So myopia is also called short-sightedness. And my way of remembering it is you could say, I can see myself. So if I'm short-sighted, I can see myself. I can see my hands in front of me because I've got short sight. That means I can see myself. But I struggle to see things in the distance. So the reason for this is that you've got too much refraction happening. For example, your lens is too thick or your eyeball is too long. And so what happens is the light focuses in front of the retina. So things that are in the distance, they look really blurry. Okay, so that's the problem. There's too much refraction. So we need to sort it out and change the refraction so that it focuses further back. So what you will do is try and spread the light out before it enters the eye. And you do that with a concave lens, which looks like this. So if the person wears glasses, they will need to use this type of a lens. Okay, the opposite then is hyperopia. So this means that people with this, they can see things in the distance really clearly. I can see that car, etc. but I can't see things right in front of me. Reading a book is quite hard. And this is because there's not enough refraction. So light that comes in is focusing behind the retina. And this could be caused by a short eyeball, so it's not very long, or a thinner lens or less curved lens than normal. So the solution for this is just opposite of concave, which is convex lens. I haven't really come up with a way to remember the convex. I mean, concave, the lens itself looks like a cave, but I just remember in terms of the physics that you want to spread the light out because you've got too much refraction in the first place. So I would try and remember that. And yes, everybody, this is triple. So triple only stuff. We covered a fair bit of combined stuff. So flick back earlier in the video if you want some combined only stuff. Then there's a little bit extra you need to know. There are other treatments than glasses. You can use hard or soft contact lenses. You'd probably get like a comparison sort of question if this came up. You can also have laser eye surgery. This can only be done on adults once the eye is finished forming and their sight is, stable. And finally, you can have replacement lenses, which is where they take your lens out and they put a new one in to refract the light the right amount. Or they can actually just leave the natural lens in and put another lens in to the eyeball as well. So there are some problems with that. You can get a risk of infection and maybe even what's it called? Yeah, cataracts developing, which are when the eye goes all cloudy. Okay, so that is parts of the eye ticked off. This is what we've got left on the list: cloning and the work of Mendel. You know what? I think I kind of covered that with understanding genetics. Did that a little bit out of order, so I can tick both of those off. So cloning at the moment is the last topic. So let me know if there's anything else you want me to do. But we'll go over to cloning for now. Okay, so again, cloning is a biology only or triple topic. Oh, I just remembered somebody wanted control of body temperature. Yeah, we'll do that in a second. Okay, so cloning, there are multiple types of cloning, believe it or not. There's loads of ways we can clone things. These two are specific to plants, and these two are specific to animals. So the first is tissue culture. This one's good because it only requires a very small number of cells, and they're taken from the plants that you want to reproduce, and they're grown under special conditions to encourage them to grow, and that will make you identical plants, and you can do this really easily. The issue with this is it's more expensive, but it will make you like hundreds, thousands of identical plants. So you can really scale it up, and you only need a very small number of cells to do it. So it's good for rare plants. Yeah. The next one is cuttings, which is a little bit of an older technology, but it is still a very reliable method. This is when a part of the plant is cut off, literally cuttings. It's normally dipped in rooting powder, which would be auxin, for example. That's on the biology spec as well. So dip it in some rooting powder, and then it is put in optimal conditions to try and grow a new plant from that cutting. And that, again, makes cloned plants because it's come from just one parent plant. Okay, the next two are for just animals. So we've got embryo transplants. So remember, an embryo is a little ball of cells that's in the very early stages of development. And that's really important that it is from an early stage of development because you don't want any of the cells to have differentiated yet. If the cell has started to turn into, I don't know, skin cell or spinal cord cell, then you can't really, you can't split it and make two embryos from it because it's already gone down that route of differentiating. So you'll split it in two, let each embryo develop a little bit further, and then you'll implant them into a host uterus of whatever animal that is. Okay, the last one is adult cell cloning. So let's say you've got an adult and they require, I don't know, let's say new nerve cells. They've undergone paralysis of the spinal cord. So they need new nerve cells. So you would take the adult cell from a part of the body, for example, the skin, and you would extract the nucleus from it. Then you would take an unfertilized egg cell and you discard its nucleus. So you take it out and get rid of it. You're then going to fuse the skin cell nucleus with the empty egg cell to make this. And then you're going to stimulate it electrically. So you give it a little electric shock to stimulate it to divide. And it will start to divide and actually form an embryo. And all of those embryos contain the same DNA as this adult, whoever the skin cell was taken from. It contains their DNA. So essentially, you're making a clone of the adult. The idea is that embryonic stem cells could then be used to cure the paralysis. So you could induce them to become like nerve cells and then inject them into the site where they're needed, like the spinal cord. If this is just in an animal, like a sheep, you've probably heard of Dolly the sheep, then this embryo would be allowed to develop and then inserted into the host sheep, the mother, into her uterus, and then it would become a baby sheep. And that baby sheep would be a clone of whatever adult the skin cell was taken from. So that is cloning, and that is the last thing on the list. I'm going to write down a couple of things that people have requested, or I will forget. I'm sorry, I don't want to make you keep saying what you want to go over. So we had somebody ask for meiosis. Yep, Anissa, we can cover meiosis. That's combined. We've had somebody ask for protein synthesis. Yep, Alex, we'll go over that. That's biology only. I'm afraid I don't do A-level or B-Tech yet. I have taught A-level, so I could do that, but I want to get through all GCSE stuff first. Safia, genetic modification. No, we haven't actually gone over genetic engineering, but I'll add that to the list. I'm doing Q&A, Majid, so you can ask me anything. I'm not going over the whole content. It's too long. If you wanted to go over it as fast as possible, watch my video. It's an hour 42, but someone has smartly recommended you can do it on double time to really pack it in. What I'm going over is requests. So these are things people have asked me on Instagram, and these are the extra things that I've covered at the moment. Megan, I'm afraid I don't have any predictions. I think it's too early to be making predictions on a new specification. There's only been 1 year. There's still so much that could come up, so I'm afraid I can't share that. And yes, I can do body temperature. I think body temperature was asked for a long time ago, so I'm going to start with that because I think I missed that. Okay, so body temperature is triple-only content. It comes up in the homeostasis and response topic. Let me find it. Here we go. So, obviously, we need to maintain a constant body temperature for our enzymes to function properly. In humans, it's a really narrow range. 37.5 is our optimal body temperature. A couple of degrees too high or too low, and things start to go wrong. So we have a thermoregulatory center in our brain. It's actually called the hypothalamus. I don't think you need to know that. I think you just call it the thermoregulatory center. And it's got special receptors that are sensitive to the temperature of our blood. So our blood is going everywhere in our body to supply all the things that our cells need. So that's a good way of recording our body temperature. Your skin cells also have got temperature receptors, and they will use electrical impulses to get a message to the brain if some external influence like flame or just being somewhere really hot is affecting the body temperature. So, of course, if it gets too hot or too cold, we need to try and fix our body temperature. So if it's too hot, vasodilation will happen. And this is when the blood vessels that supply the capillaries on the surface of our skin, so right up at the top of our skin, that's when they widen. Dilation just means widen. When we talked about the eye, we could have said that the pupil dilates, which means it widens and gets bigger as well. So that's what the dilation part means. And vaso just means blood. So vasodilation, widening of the blood vessels that supply the surface capillaries. Now that's important. Please don't say that the surface capillaries vasodilate, because they don't. Capillaries are only one cell thick, one blood cell thick. They hardly let any blood through. Well, they're a little bit thicker than that, but anyway. They are not capable of vasodilating or vasoconstricting, but they are capable of having more blood pumped through them at a quicker rate. So it's the ones that supply the capillaries that vasodilate. Okay, so more blood traveling along the surface of the skin means that more energy is transferred to the surroundings by radiation. So there is heat loss, and that means your body temperature will drop back to normal. Then you also are going to sweat. You definitely will have experienced this. You're going to release a lot of sweat, and that is going to mean that the water in the sweat will evaporate. And as it evaporates, energy is transferred to your surroundings. So that's the second way of losing heat from your body. So just make sure with this one, a really big tip is make sure you just include that last bit of saying energy is transferred to the surroundings from the skin or from the body. That's really important to get the mark. Okay, if it's too cold, then all of these things happen. So vasoconstriction is literally just the opposite of vasodilation. Instead of the blood vessels widening, they narrow, so they become really thin. And that, again, is still the blood vessels that supply the surface capillaries. So there's going to be less blood passing through those surface capillaries, and therefore less energy is lost and transferred to the environment or surroundings. Also going to be less sweating, so that less water evaporates from the skin, losing energy to the environment. And then finally, shivering will happen. So if it's really cold, your body's going to involuntarily start to shiver. And this is just the rapid contraction and relaxation of your skeletal muscles, which are all over your body. And that is going to mean that loads of respiration is happening, because respiration provides that energy for movement. But as an extra point, the respiration releases heat or thermal energy, because hopefully you remember from bioenergetics, respiration is exothermic. So it's releasing heat. It's going to keep your body temperature at the right level. It's going to increase it to keep you warm. Okay, so that is everything you need to know for control of body temperature. So we'll tick that off. Okay, Coral, yes. Will protein synthesis come up? I don't know. Absolutely, it could. It's on the specification, but so could everything else. So I am going to go over it. Yep, it's on my list. We'll just go over meiosis first, because that's written above it. Nice to have an order. So meiosis is on inheritance topic. So let's find that. So the first thing is meiosis is used to create gametes. Gametes are just your sex cells. So your male sex cell is your sperm. Your female sex cell is your egg. So meiosis makes egg and sperm. So what happens is the exact same things that happen in mitosis up to this point. So we'll recap that. The parent cell will start by doubling the genetic information or copying it. So in this case, the parent cell has 2 chromosomes in its nucleus. So it will double them so that you end up with 4 chromosomes. And then it will go through a first division. So it splits in 2. So we're back to having just 2 chromosomes in each cell. And that's what mitosis does. The rest of this is meiosis. It's really simple. There's just a second division. So in the second division, the chromosomes are split again. So you've now only got 1 chromosome in each cell. So the differences between meiosis and mitosis are, first of all, in the purpose of them. Mitosis is just for making new cells for growth and repair, but meiosis is for making new gametes. The other difference then is the number of cells are produced. Meiosis makes 4 cells, and mitosis just makes 2 cells. And then finally, the third big difference is meiosis halves the number of chromosomes. So if you compare the parent cell to the daughter cells, there's half as many chromosomes. In mitosis, the daughter cell would have the exact same amount of chromosomes as the parent cell. So that's it. That's meiosis and a little bit of mitosis covered. Let me know if that makes sense, if you've got any questions on it as well. Okay, but if not, I will tick it off and we'll go on to protein synthesis. For anybody that's just joined, let me know what else you would like to cover. This is my long list of requests. We've done most of them and we're on our last few now. Okay, so protein synthesis. That is another biology-only topic. It requires you to understand the structure of DNA, so we'll do a quick go on that as well. Have some water if you haven't had some in a while. Good to be hydrated for your exam. Okay, so protein synthesis and DNA structure. Let's find that. Okay, right. I should try and cover up part of this because it looks a bit scary when you see it all at once. Let's start with DNA structure. You do need to know a little bit of DNA structure if you're doing combined science, but all you need to know for combined science is that it's a double helix and it's made up of repeating units, so it's called a polymer. So it's a polymer that forms a double helix. That's the combined science stuff. And now we're going to go over all of the higher tier biology only stuff. Yasmin, I've already done Punnett squares. If you flick back probably quite a bit, maybe in the first half hour or so, I think I covered that then. Okay, so triple only, you need to know more detail. So all these little colored things that are going between our 2 strands, those are bases. And there are 4 bases. We've got A, T, C, and G. So A always joins to T, and C always joins to G. My way of remembering that is a little tip that my brother told me years ago. He loves golfing, so his way of remembering it was at the golf club. So A and T, they always go together. Golf club, G and C, they always go together. So you need to know that. Okay, and then you need to know what one of the units is. These are all repeating units, and we've got P for phosphates, S for sugar, and then this is your base, which is one of these 4. So this unit is called a nucleotide, and you have got that thousands and millions of times in your DNA. So it's just loads and loads and loads of them all joined up together. So you'd have another nucleotide down here and down here and down here and above and so on. So that's it. That's what you need to know about the structure of DNA. Now we're going to look at the protein synthesis side of things. So you should hopefully already know that genes code for proteins. And even for combined science, you need to know that. You also need to know that genes code for amino acids, which then make up the proteins. So a gene is just a short section of DNA. Okay, and this is how it works. So we're starting in the nucleus, which is in here. And we've got our DNA strand here. The DNA is used to make a template, and specifically one of the genes. So let's say it's this gene. This gene would make this pink template. You don't need to know any more detail than that. If your teacher has given you more detail than that, you can forget it, because that's probably A-level standard. So the template is made. It will then leave the nucleus. There are actually little pores. Pores just mean like holes, like the pores in your skin. So it leaves the nucleus through a pore and travels into the cytoplasm. And then in the cytoplasm, it will come along and it will join onto this thing, which is a ribosome. So the template attaches to the surface of the ribosome. So they're kind of stuck together right now. So that's step 3. For step 4, a carrier molecule will then carry obviously, carry an amino acid along to the template strands. So I've used little squares to represent the carrier molecules. And if I hold this a bit closer, you can maybe see that that says AA for amino acids. So here we've got 2 template molecules and 2 amino acids brought beside each other. So they are brought along onto the template strands, and they are brought in a very specific order that has been decided by the DNA. So the template strand is sort of making sure that those carrier molecules line up in the right order. So if I zoom in over here, I've kind of got a bigger picture of this. This is our pink template strands, and here are 3 different carrier molecules. They bring the amino acids into the right order beside each other, and the amino acids will then join up, and they're actually forming bonds between them to make a protein. Because remember, proteins are just long chains of amino acids. So the loads of these will join up and make a protein. Once the carrier molecule has brought the amino acid over to the template in the right position, it releases the amino acid, and it is then free to go back into the cytoplasm. And, oh, can't really read that. And it will pick up another amino acid. You still can't read it. Sorry, that's bad. Let me just unfold the paper for a minute. Maybe you can see it now then. And it will pick up another amino acid. So that is protein synthesis covered at GCSE level. I hope that helps. Let me know in the comments if it makes sense, and take a screenshot if you would like to. I have offered to also take some photos and stick them on my Instagram stories, but I know those are pretty small. So yeah, you can take a screenshot. If you're watching on a laptop, it's probably quite nice and big. Not sure about the quality, but hopefully it's okay. Alex, I'm afraid I haven't made any videos on chemistry and physics. It's on my to-do list, but I want to finish biology first because I only did paper 2 in time for these exams. And then I'm going to do chemistry and physics for next year, but I'm afraid it's too late for you. But yeah, I can teach them. Okay, thanks RX Alliance for checking out my nostrils there. Yeah, getting very up close and personal. Okay, so that is protein synthesis. The very last thing I've got on my list is genetic engineering, which is combined science. And then if there's anything else, stick it in the comments. There's a chance I have missed it. Oh, Grace, you asked a question. Do we not need to know about mRNA? No, you don't. That is A-level. If you're studying AQA specification, then that word doesn't need to be in your vocab. Okay, so genetic engineering is next. And then Yasmin, you want to do, I think you mean reaction times. I've actually already covered that require practical, so you can take a look a little bit further back in the live stream if you want to do that. Okay, genetic engineering is in the inheritance topic. Let's find that. Here we go. Okay. Oh, and somebody else had also mentioned about wanting to know a bit about why it's done. Okay, so first of all, why do we do it? Well, the big benefit of genetic engineering is that we're going to make genetically modified crops. And these crops can have loads of great characteristics. So, for example, they can be resistant to disease. They can be resistant to insect attacks. So they could have slightly thicker, waxy cuticles so that the insects can't bite and eat them, things like that. They can have an increased yield. And yield just means that when you're growing your crop, you can get a lot more of it in the same space. So a farmer is going to get more crops for the same size field. They can make bigger or better fruits, which obviously sell for more money. And they can be herbicide resistant. Herbicide is used to kill unwanted plants. And so if you make your crop resistant to it, you can spray your whole field with the herbicide. Your crop will still be there, but all the other plants that are there will die. Okay, so that's why we do it. People do have some concerns with it. Some people think that GM crops might actually harm human health. So far, that hasn't been shown to happen. So yeah, no real risk, but it is a concern. We're not sure for everything. What's this say? Oh yeah, modified genes could spread. So the classic example is that some GM crops were made to be infertile. If that spread to a wild population of plants, we could end up accidentally causing their extinction, or at least wiping out some communities of them. So we want to make sure that the GM crops don't spread in an uncontrolled way. But again, so far that hasn't happened. And then finally, whenever we are modifying the genetics of organisms, people are often concerned that this could go too far and might cause a desire for designer babies, which is basically just when you choose the characteristics that you want your children to have. And that's, some people feel like that's playing God. But try and use the phrase designer babies, because that's what AQA talk about in their specification. Alex, I don't think I will have A-level biology covered for next year. Like I said, I want to cover all the GCSE stuff first, and it does take a while for my videos because I have to make all the drawings and things. But at some point in the future, it's a 2-year course, so you never know. I might have some made by the time you're finished, or at least get in there. Okay, now let's look at how do we genetically engineer organisms. So the first thing, if you're studying foundation, is that you are going to cut out the parts of your chromosome, the gene, that gives you the good characteristic. So let's say, very simplified, you had a gene that makes a cow produce loads of milk. It's just one gene. That's not true, but anyway, let's say we had that as a gene. You might cut it out and you want goats to have this gene as well. I don't know. You would then take that gene and insert it into the chromosome of the organism that you want to have that characteristic. And then whenever you allow that organism to develop, this would be like in the embryo stages, it should have that desired characteristic. So that's the foundation level explanation. If you're studying higher tier, there's a little bit more to it. You need to know the name for the scissors, and that is enzymes. So I'll write that down in case you want to screenshot this in a moment. Enzymes are used to cut the chromosome and remove the gene that is desired. The classic example is the insulin gene. Obviously, we want to treat type 1 diabetes with insulin injections, so we need to have lots and lots of insulin generated. Can't extract that from humans, so we're going to have another way to do that. So what you're going to do instead, I'm just writing some more words down, is you're going to take that insulin gene and you're going to want to put it in something else. So over here, we have got a bacterial cell. This is one example of a vector. You can actually also use some viruses as vectors, but here's our first example. So this is a plasmid. So if you remember from topic 1 of cells, a plasmid is just an extra piece of DNA that is found in a bacterial cell. They've got their main DNA in just a little free-flowing loop over here, but this is extra chromosomal DNA. So the plasmid is extracted, is cut with the same enzymes that were used to cut your other chromosome with your insulin gene from a human, and it's opened up, and then the human insulin gene is inserted into the plasmid. It's sealed, and you need to check that it's sealed, and then it's inserted back into the bacterial cell, and then you will monitor and make sure that the bacterial cell has taken up the plasmids, and then you're just going to allow it to reproduce. And when it does, every single bacteria that it makes will have that insulin gene, because remember, bacterial cells reproduce asexually. So now they've all got that insulin gene, it means that all of them are going to be able to produce insulin, which is that hormone that regulates blood glucose levels in humans. And then once they produce it, you just need to extract it and purify it, and then you can use it, which is really, really great. It's made a huge difference to the life of people suffering with diabetes. Okay, so that is the process of genetic engineering. And I think that's all of it. Okay, so you're really welcome, Coral. If anybody's watching and wants to say, thanks, Emma, just click subscribe, because it will really help me get my videos out for next year's GCSE students. So hopefully they can do really well and be confident for their exams. Okie dokie, so is there anything else that we would like to know? So I've seen some questions. Ellie, you've said, is there a use of genetic engineering involving sheep and human drugs? Quite possibly. I'm not sure. Well, there isn't one explicitly on the specification that you need to know about, but I'm sure there is. Maybe you can tell me more about it. Yasmin, can I go live for physics and chemistry? The truth is that I would need to sit down myself and do a lot of revision if I were going to go live for those. Despite having taught them, I haven't taught them this year, so I would need to do a lot of my own revision, believe it or not, for those papers to help you. So I wouldn't feel like I probably would be able to help you to the best of my ability. So I probably won't, but I think there are some other YouTubers that will be doing lives for that. So I hope that you do find somebody to help you with those. Okay, let's see what else. Is there anybody else anyone wants to go over? So Aisha, yes, I have already covered protein synthesis. It was pretty late on my list of requests, so you shouldn't need to go back too far to find that. So take a look. I did hold up a diagram with it, so maybe if you scroll back and look for me holding it up, well, I've been holding up my paper a lot, so you might struggle. Yeah, so have a look and see if you can find that. Is there anything else anybody wants to go over? Maybe I will just show you a little overview of some of the topics, and then you can see if there's anything appearing on the list that you don't remember. Thank you to everybody for your nice, kind comments there. Okay, so I'll start with the homeostasis topic. This is everything that is on it. Is there anything anybody wants to go over from that list? Aisha, you said eutrophication. That really doesn't come up explicitly in the AQA specification. It's very brief. I'm just wondering where, even if that does come up, a couple of you said it. I haven't done a video on that. Is that triplet only, or is it kind of buried in the spec? I have got the spec in front of me. I've been telling people that the best thing to do if you're not sure if something's on the spec is you just do Command F or Control F, and it pops up a little finder window, and you just type in your word. You got to spell it right and press search. And I've just done it for eutrophication, and it says 0 results, which means that word isn't on your spec, as far as I'm aware. Can anybody tell me where they think they've been taught it? Because I don't think it is on the AQA spec, so you can probably leave that aside. Okay, Laura, you've said reproductive hormones and diabetes. Okay, so I've already covered reproductive hormones, but I haven't covered diabetes. So let's go through diabetes. You said you've been taught it in triple. Yeah, well, I'm not sure at what point you could have been taught about it briefly in relation to decay. I'll mention it, but it's not explicitly on the specification. So in the decay process, obviously organisms break down, decomposers break down dead organisms. This is an issue when eutrophication happens. So eutrophication is when there's surface runoff from fields of fertilizers. So fertilizers get into our waterways and they make it really nutrient rich. So suddenly we get a huge algae bloom, which is all the plants in the water. They get loads and loads of nutrients, so they grow really rapidly and they cover the whole pond, and yeah. But that doesn't last forever. So when the nutrients run out, eventually the plants will die. And as they die, decomposers come along and break them down. And as they break them down, they release carbon dioxide. But more than that, they use up the oxygen, because remember, all living things respire. So if they use up the oxygen from, let's call it a pond or a lake, then there's no more oxygen left for the aquatic organisms that live there, like fish and seals and things that live under the water. So unfortunately, you get death of all of those organisms that live in those waterways. So that's what eutrophication is. It doesn't seem to be explicitly on the specification, but just if you wanted to know what that was. Okay, land and water pollution. Yeah, so actually I have got a little bit on land and water pollution. The main things that you need to know is that toxic chemicals can wash into waterways. So yeah, I don't think you need to know a lot about eutrophication. If it did come up, it would probably come up with a little bit of context or information to use from other parts of the spec. But I don't think you'd be expected to use that word in your answer since it's not in the AQA spec. Now I lost my train of thought. I was going to look up something there that somebody asked about. What was it? Oh yeah, diabetes. Okay. Diabetes comes up after control of blood sugar. So I actually have a blank table. I have done live streams for each of the topics in this paper, but we didn't cover this when we did it. I only did whatever people wanted me to do. So it's blank at the minute. So let's talk about it. So you need to know what the disorder is. So basically what's happening in the body to cause it. So in type 1 diabetes, the problem or the disorder is that the pancreas does not produce any insulin. So it's not making it naturally. The cause of this, we think it has got a genetic element to it because it's often something that young children develop. So you will know if you have it by normally very early teenage years. It's kind of the latest it would be discovered. So we think there's a genetic element to it. Because the pancreas doesn't produce any of its own insulin, the treatment is to take insulin injections. So people with type 1 diabetes will often inject themselves before a meal. You probably know somebody at your school that has type 1 diabetes, or maybe even you've got it. And you'll see them injecting themselves before they take a meal. They'll also try and control their diet to make sure that they're not putting in too many carbohydrates, which get broken down into sugar or glucose, and not too many sugary foods themselves. Another way that they can help sort of control their glucose levels naturally is with exercise, because when you exercise, you use up glucose in respiration. Okay, so that's type 1 diabetes. Type 2 diabetes is a disorder that is caused whenever the pancreas produces insulin, but the body cells no longer respond to it, which is bad news. It means that even though that hormone is there, the cells aren't responding and converting the glucose into glycogen like they should be. So that's the problem. The causes of it, there are 3 main risk factors that you need to know. The first is obesity. So being very overweight can increase your risk of type 2 diabetes. The second is a lack of exercise, which sort of goes hand in hand with being obese. And the third is old age. As you get older, your risk of type 2 diabetes increases. So the treatment is pretty straightforward. It kind of lines up with what the causes are. The first one is going to be weight loss. If you're obese, you can reduce your weight to try and help with that. The second is increasing exercise. And then the big one, just as I mentioned before, but is way more important, is carbohydrate controlled diets. So they need to make sure that they're not having too many carbohydrate rich foods in their meals, because that spikes their blood sugar and they don't really have a way of bringing it back down. If it's an extreme example, they may be given some form of drugs to try and control it, and they could even revert to giving them some extra insulin to see if that could work. Okay, so that is diabetes and how it is treated. The kind of question you're going to get with this is potentially a comparison, or it's going to be to perhaps interpret and come up with some information with normally relation to treating type 1 diabetes. There's a lot of new monitors that have been devised. So it's a way of, for example, constantly regulating the blood glucose levels. In the well, not just the old way, because a lot of people still use it, but they have to do like a finger prick test, and they test tiny sample of their bloods very regularly. But a lot of new treatments that have come out are using automatic sensors to detect the blood glucose levels. So you could get some information asked to explain why that method is better than another method, something like that. Okay, let's see now. What else do we want to go over? Can you go over photosynthesis? Jessie, I'm guessing you might be at Excel, because for AQA, photosynthesis is on paper 1. So I'm going to leave it because I think most people are here because this is for AQA. Oh, I need to go back a little bit because we've had quite a lot of comments there. Majid and Yasmin, I've already gone over the carbon cycle and meiosis. Unless some other people want that again, I will leave it for now. Let's see, can I go over cell division again? I've already covered that. Do I have any good predictions? No, I'm not going to make any predictions. As I've said before, I think it's too early on in the new specification. There's only been one paper, so I'm not going to make any, I'm afraid. Could I go over treating infertility? Aisha, I already have, but I can go over it again if we're done with anything new, if there's nothing new that comes up. And Hayder, I've already gone over the quadjet practical, but if there's nothing new, then I will go over it again for you. RX, can I shout you out? I just did. Okay, here's a new one. So we'll start with this. Can I go over the biogas and then the decay practical? So I've done the decay practical, but biogas I haven't done. Oh, and then contraception. Yeah, I haven't done that. Okay, so biogas and then contraception. I'm going to write that down or I might forget. Okay, so biogas or fermentation and that sort of stuff, that comes up in biology only stuff. So if you're studying combined, you don't need to worry about that. Okay, so it's very brief in the GCSE biology specification. Hang on a second. Let me just see where I've got it in the notes. Yeah. Do you know, I'm not even sure I've got a section on biogas. Let me see. So I've got this on the roles of technology, or sorry, biotechnology. With the biogas, I don't think that even comes up, do you know, in the biology only spec. I think it used, biofuels come up very briefly, just in relation to deforestation, because we are cutting down trees, particularly in tropical areas, to grow crops for biofuel. But biogas, I don't think that comes up. Sorry, who was that asked? Eclipse. I'm not sure about that. Can other people comment? Have you come across biogas? Do you know what part of the spec that's meant to be on? Because I don't think it's on the spec. So actually, let me know. There's biogas generators very briefly. Yeah, most people are saying they haven't. And maybe it comes up in the textbook. Sometimes things come up in the textbook as like extra information just to kind of fill out the specification, give it some meat. But I'm pretty sure it's not actually on the spec. Once again, I will use my little control find trick and just type in biogas. Aha, we've got one sentence on it. Okay, so it actually comes up under decay, or rather decomposition. So here's your one sentence that you need to know about biogas. Anaerobic decay produces methane gas, and biogas generators can be used to produce methane gas as a fuel. That's literally all it says. There's no extra information saying a student should be able to explain or anything like that in relation to biogas. So I think that's it. Hopefully you'll be okay with that. So biogas generators can be used to produce methane gas as a fuel. And the good thing to know is that methane gas is a very clean fuel. You might have covered this in chemistry as well, but when it burns, there aren't any impurities to make, for example, sulfur dioxide, which causes acid rain. So, okay, so that's biogas. And I said I was going to look at contraception, and then we have some more questions coming in. A lot of people wanting to go over the menstrual cycle again, and negative feedback as well. So I will just note that down, and then we'll do contraception. I haven't got a whole section on negative feedback. Negative feedback sort of comes up across the spec in a few places. We've got lots of questions coming in now. Okay, everybody wants menstrual cycle. Right, we're doing that after we do contraception because somebody else requested that first. Okay, so you need to know the different methods of contraception. You could definitely get asked about evaluating how good those different methods are. You'll get given some information, most likely, but you should know some general advantages and disadvantages of each. Okay, so types of contraception can be split into hormonal and non-hormonal. So hormonal ones are going to control the female reproductive hormones. So any oral contraception, also called the pill, is going to inhibit, that means stop, FSH. And if you remember, both estrogen and progesterone do that. So most pills contain both of those hormones. Sometimes you get progesterone-only pill. But anyway, they both inhibit FSH, which basically means they stop FSH being produced, and FSH matures the eggs. So without it, there's no mature eggs, and therefore you're not going to be able to get pregnant. So that's the first method of contraception, and it's hormonal. The other hormonal forms of contraception are the contraceptive patch, the contraceptive injection, and the contraceptive implant. So the patch is just placed on the outer part of the body, on the skin somewhere, and it will release the hormone again to inhibit FSH, and that's progesterone this time. So all of these use progesterone. It will need to be replaced. I think it normally lasts about a week, but it's very short term. So if you're experiencing side effects, it's easy to remove. Then there's the injection. You will need to go and see a nurse or a doctor to get that. So you need appointments. And then there's the implant, which again, a nurse or a doctor will insert. That goes under the skin, normally somewhere in the arm, and it will stay there for, I think, something like, can't actually remember. I think it's like 1 to 2 months. It's in my video. I forget the exact details, but you don't need to know the exact details. So progressively, they last for longer and longer, and that's a good thing because you don't need to remember to take them. The pill is something that needs to be taken every day, so there's quite a high risk of human error and therefore an increased risk of pregnancy. Okay, a question here, where is FSH released from? It's from the pituitary gland in the brain. Aidan, I'm afraid estrogen is the one that's produced by the ovaries. I remember FSH is produced by the pituitary gland because FSH sounds like fish. It's just missing the I, and fish can swim all the way down from the pituitary gland to the reproductive system where it acts. So yeah, it's that. Okay, don't worry, it's hard. Okay, so that's the hormonal side of things. Now we've got non-hormonal. So first up are spermicides. So this is something that is able to kill the sperm, and it's, what's the word? It's kind of like a fluid, but like a jelly-like fluid, I think. It's really unreliable, so it should only be used in combination with another form of contraception. So that's a really unsafe method of contraception on its own. Then you've got the barrier methods, which include condoms that fit over the penis, and then you've got the diaphragm that acts as a barrier into past the vagina into the uterus. So that makes sure that the sperm can't reach the egg. So both of these will stop the sperm from reaching the egg. Some benefit is, for example, that they help prevent some STDs or sexually transmitted diseases because they stop the sharing of those bodily fluids. So that's a real benefit. They're also quite inexpensive. You don't need to go and see a doctor to use them. But the disadvantages are that they can be often used or fitted incorrectly. And if that happens, then there is a risk of pregnancy. I've seen some questions. What type of questions would come up about contraception? That is going to be questions like, Well, you could, in foundation papers, you're going to maybe be given some pictures and asked to identify which one's which, or you could be asked to name a barrier method of contraception or name a hormonal method of contraception, something simple like that. Harder questions are going to be things like asking you to compare the effectiveness. Quite possibly, you'll be given some data to interpret and evaluate. So evaluate means you have to weigh up the pros and cons. So you should give advantages and disadvantages of as many of them as possible. And often you'll be given some data to support your arguments for that. So that's the type of questions you would get for that. Okay, so next up, for non-hormonal, we've got the IUD, which stands for intrauterine device. So it means this is like a little, it's quite small, looks like a T. It gets inserted into the uterus. You will need to see a doctor for that. And it can be quite uncomfortable, and there's a small risk of infection when it's first inserted. But once it's in there, it lasts until it's taken out. So a really, really long time. And you don't have to think about it, so there's no risk of forgetting. So that's a real benefit of it. How it works is it can prevent the implantation of an early embryo. In some religions, that wouldn't be acceptable because fertilization would have taken place, the egg and the sperm would have met, started to become an early embryo, but then the early embryo just can't implant. But other ways that it can work is some of the IUDs actually do have the hormone progesterone. So those ones are stopping mature eggs from forming in the first place. So two kind of different ways you can have IUDs. Then there is surgery, which is permanent. So that would be whenever the sperm ducts are cut and tied, so that sperm will actually not get released from the penis during intercourse. And you can also have the oviducts be cut and tied as well. But you need to be obviously really sure that you do not want to have any children, because it's fairly permanent. I think you can potentially undo it in certain cases, but it's not guaranteed. And then finally is abstinence, which means not having sexual intercourse when the egg is in the oviduct, i.e. shortly after ovulation. So that's an extremely risky method. It basically means you're predicting the days of the month when you shouldn't be able to get pregnant, and only having sex on those days and avoiding the days when the egg's in the oviduct. That's really highly risky. Bodies aren't like clockwork. The egg doesn't always get produced exactly on day 14, and cycles, yeah, they're hard to predict. So very risky methods. Okay, so that is the sort of overall gist of contraception. Let me see if we've got any more questions. Just to make sure, only condoms can prevent STDs, right? I'm not sure, actually. Diaphragms, I think there's probably a higher risk of contracting an STD with them. So I don't think either of them are 100%. It depends. I would have to look into it, to be honest, Hader. If you want to look into it, I recommend the NHS websites. They've got a lot of really good information on the different forms of contraception, and they would probably cover that on there. Okay, any other questions? What's the difference between the copper IUD and the other one? So I think the copper one is the one that prevents the implantation of the early embryo, and then the hormonal one is just releasing progesterone very steadily to keep that level of that hormone constant so that no FSH gets produced and therefore no mature eggs get produced. What else? What is most likely to come up, Edward? I'm afraid I have no idea. I'm not going to make predictions. We've only had 1 year of the new specification, so sorry, I can't. Okay, next up, we had so many people ask for the menstrual cycle again. I have covered it, but I know there's a lot of new people have joined, so I'm going to recap that now for everybody that's asking for it. If there's anything else you want covered, please stick those into the comments, and I'll try and work my way through them. Okay, so there are 2 levels to the menstrual cycle. If you're studying foundation, you do still need to know the 4 hormones, but you only need to know a little bit about what each one does. So we'll start just by mentioning the overview of the menstrual cycle. It is a 28-day cycle that happens in the female reproductive system. On day 14, ovulation occurs, which is when one mature egg is released from the ovary and travels into the oviduct. If it gets fertilized, it will become a fertilized egg and implants into the uterus lining, which is built up nice and thick, ready for its arrival. But if it doesn't get fertilized, then the uterus lining will just break down and it will leave the body through the vagina. And that's also called a period. So that happens on days 1 to 5 of the cycle approximately. So that's the overview of the cycle. Now we're going to look at the hormones that are controlling it. So first off, we've got FSH, which I've just been mentioning quite a lot, and it causes eggs to mature in the ovaries. And it will try and cause just one egg to mature each month. When that egg is mature, then LH, that's our second hormone, LH will cause the egg to be released, which is also called ovulation. So LH is responsible for that. Then estrogen and progesterone, they do the same job for foundation at least, and they both are responsible for maintaining the uterus lining. And that means that they keep it nice and thick so that it's ready if the egg does get fertilized. So if you are foundation level, I'm going to hold this up and you can take a little screenshot, and that's all you need to know if you are foundation level. Also just on the side, got a little bit about the male and female reproductive hormones. The main ones are testosterone, which is produced in the testes for males, and for females, it's estrogen, which is produced in the ovaries. And those hormones cause your secondary sex characteristics, which is basically the changes that take place during puberty. Okay, so that is, that's reproductive hormones and the menstrual cycle at foundation level. Now let's swap on to higher tier. Okay, so, sorry about that, Majid. Can everybody else hear? Hopefully. Let me know in the comments if you can't hear. Okay, that's good. Really sorry about that, Majid. I'm not sure what's going on. Okay, so higher tier is quite a lot more you need to know. So FSH is produced in the pituitary glands, and it's going to cause the eggs to mature, but it's also going to stimulate estrogen production. So we're going to learn about how the other hormones interact with each other. So it stimulates estrogen production. Estrogen, as we mentioned previously, is going to help with the uterus lining. So it's going to start building up the uterus lining, which means it gets a better blood supply and it becomes thicker. It then is going to inhibit FSH. And inhibit just means that it stops the production of, because we don't want more than one mature egg each month. If we accidentally did have more than one, you could end up with multiple eggs in the oviduct and have a risk of multiple births if they get fertilized, which is risky for the mother and the baby. So our bodies naturally try and stop that, so FSH gets inhibited by estrogen. But it then stimulates LH. So LH, remember, carries out ovulation. Now, that's actually all you need to know for LH. There's no extra interactions there. And then finally, we've got progesterone. So same thing as before is that it's going to help maintain the uterus lining, but it's also going to impact our other hormones quite a lot. So it's going to inhibit both LH and FSH. We don't want any more ovulation happening, and we don't want any more mature eggs happening. Okay, the last thing is just to mention where it is produced. So it gets produced by the empty follicle that's left behind in the ovary when the mature egg leaves the follicle. So it's produced like in here inside one of those little eggs, or rather the shell, the follicle of the egg. Okay, so again, if you're studying higher tier, you can take a little screenshot of that. I'll try and hold it steady if you want it. Thanks, Hayder. Good luck to you as well. I know you'll be doing great too. Okay, so that's the menstrual cycle. Okay, so anybody who's just joined, we've covered loads and loads and loads. Thank you, everybody who just answered Coral's question. So this is what we have covered. It is a huge, massive list right now. Wow, look how much we've done. This has been a rather long live stream. but I can see there's lots of you that want some help, so that's really good. Okay, we're down to here. Negative feedback is the last thing on my list. Yay! You're really welcome, Hader. To everyone that's saying thank you, if you want to help me, please subscribe. I've said before, but it will help my videos get out there for next year's students so that they can see them and do really well, and yeah, hopefully be confident for their biology. And other subjects, I'm going to do other videos at some point as well. Okay, so I have said before that I don't actually have any notes on negative feedback because it comes up on different parts of the specification. So lots of examples, but I'll explain the principles of it. So in my body, we have a lot of conditions that we need to maintain at an optimal level, which means trying to keep them constant so that they work at their best. For example, body temperature, we want to keep at around 37.5 degrees. If it gets too high, our enzymes will start to denature and our cells will not work properly because they don't have enzymes to help them. And if it gets too low, again, the enzymes will stop working because they slow down, etc. Excuse me. So negative feedback is what controls our body conditions. So essentially, if the condition deviates from the set points, for example, if body temperature gets too high, then there will be something in the body that detects that. So that's often called a coordination center, because it's going to detect it and it's going to decide what response is going to be taken. In the example of body temperature, it would be the thermoregulatory center in the brain, also called the hypothalamus. So that would detect that the body temperature has gone too high, and then it will decide what response is going to happen. And that will spread out normally through the nervous system or sometimes the hormonal endocrine system. And the response will happen. So in the situation of temperature being too high, you would start to sweat and you'd start to have vasodilation when your blood vessels supplying the surface capillaries, they widen, and therefore more blood travels through the surface and more heat is transferred to the environment. So that will then reduce the body temperatures, bringing it back to the set points. So you can see it's a chain. If the opposite happens and it goes the other way, so if body temperature went too low, then a different response would be produced to bring it back to the set levels. So that is, that's negative feedback. It's a loop. You're definitely going to get it in relation to some examples, or if it were a 6 marker, it could be something like describe examples of negative feedback and what this is, and you'd have to choose some examples from the specification. That would probably be in biology only because you've got lots more examples of negative feedback. But it could come up and combined as well for higher tier students. Okay, so question mark person, I have literally just done it. So maybe if you go back like 5 or maximum 10 minutes, I think that should come up. Grace, you would like to go over trophic levels. Okay, so that's a biology only topic. So I think they're quite nice, trophic levels as a topic. Essentially, there are 4 trophic levels, and we just call them 1, 2, 3, and 4, but there are other names. So trophic level 1 is always producers. So these are things like green plants and algae. So just remember, it can also be algae, it's not just plants. And they are able to produce their own source of food using the sun's light energy. So basically, they do photosynthesis. So that's trophic level 1. Trophic level 2 is above it when we do a pyramid of biomass, and that is a primary consumer. So those eat producers. Trophic level 3 is secondary consumers, which eat primary consumers. So these are predators and these are prey. And then finally, trophic level 4 is our tertiary consumer. This is normally called an apex predator because they don't have any other predators above them hunting them. Occasionally, food chains do go one step further, but it's pretty rare because, as you can see, the biomass is shrinking hugely. Lots and lots of biomass is being lost, so there's less energy to pass on to the level above. So that's why food chains don't often go beyond these 4 stages. So that is kind of it. The next, just to mention on that point, is that only about 10% of the biomass, even from producers, is passed on to the next level. So 10% pass on, 10% pass on, 10% pass on. So you lose so much biomass at each level. Yeah. There's another little side note here, and it's about plants and algae. They only actually transfer about 1% of the incident energy of light. That basically means the light that makes it through to our planet Earth. They only transfer about 1% of that in the photosynthesis process and help use it to turn carbon dioxide and water into glucose. So it's pretty, it's pretty small amount of light that actually gets used for photosynthesis. Okay, so thank you, Safia. I am actually going to finish up really soon because I do need to have some dinner and go for a walk, and so should you. Make sure you take a break. So let's see, what time is it? 7:30. Whew. Right, I'm going to say 10 more minutes. If there's anybody that really is having a little bit of a panic, I might do a last minute live stream tomorrow morning, tonight. I'm not sure, but I think I've covered most of it. And I have got a lovely video. It's an hour and 42 minutes long, so it's not too long. So you could watch that this evening if there's any last bits you want to go over. And you can even do it at double time or double speed to help you watch it faster. So last topics then. Let's see. Endocrine system. I could do that very briefly. Hader, I have covered that in quite a lot of detail. So it's further back in the live stream. Different types of mutations. So Alex, you actually don't need to know all the different types. That's A-level content. All you need to know is that mutations happen continuously. So they're happening all the time. Most of the time, they go completely undetected because they don't actually change something significant. So they don't affect gene expression or they don't change our phenotype. And actually our DNA can sometimes repair mutations too. So, but occasionally they can. So if a mutation happens in the DNA that causes a different amino acid to be brought along whenever we're doing protein synthesis, then that can actually change the end product of the protein, which then changes some characteristic, whether that be like, eye color or antibiotic resistance or something like that. So yeah, but that's it. If you've been taught things like substitution and deletion mutations, that's actually A-level. That doesn't come up in GCSE. Sexual and asexual reproduction, Ellie, a million question marks. I think you've asked for that before. So yes, let's cover that very briefly. So sexual and asexual reproduction is on combined science, and then advantages and disadvantages of it is also on biology only. No, Alex, you don't need to know any of those, not at all. You can check the spec, but it's not on there, which is good. Okay. Sorry, I'm trying to find this. Sexual and asexual reproduction is on inheritance. So it should be right at the start. Oh, do you know what? I haven't made any notes on it because I think it's something I can talk through quite easily. So sexual reproduction is where there is fusion of gametes. Now in animals, this is the egg and the sperm. But in plants, it is the pollen, which is the male gamete, and an egg. So just be aware that it's not always egg and sperm. There's also pollen and the egg in flowering plants. So sexual reproduction produces variation in offspring. Because there is fusion of gametes, and gametes are each unique, meaning that there's no two that are the same, we get a huge amount of variation in offspring. So I'll talk about the advantages of that. That's in the biology only part, and the disadvantages. So the big advantage is that having variation in a population can allow it to undergo. My neighbors have just started drilling. I think that's a sign I need to finish the live stream. Yeah, it's a good thing because they can undergo natural selection. So if the conditions change and there's a lot of variation, some of the organisms in that population might have adaptations that allow them to survive in those new conditions. And over a long, long period of time, natural selection will happen, causing evolution of that species. A big disadvantage is it takes a lot more time and energy to carry out sexual reproduction because they have to find mates. So animals have to find a partner to reproduce with. And also the process of it is a lot slower. So, I mean, pregnancy in humans, it takes 9 months. In some elephants, it takes like 2 years. So it's a lot slower. Okay, so now asexual reproduction. So this is whenever there is just one parent, and that parent will produce their offspring using mitosis. Remember, mitosis is going to produce daughter cells that are identical to the parent cells. So it's going to produce clones of the parent plant. So that means there is no variation, as there was no mixing or fusing of genetic information. In some cases, that's a good thing. So for example, if the conditions were favorable, if there were good conditions, then a lot of reproduction can happen really rapidly. And so you'd have loads and loads of your identical offspring all surviving really well. The bad thing is if the conditions change, then there's no variation to allow natural selection to occur. But on the flip side, another good thing is they don't have to spend any time finding mates, and it's really quick. Some bacteria can reproduce in as little as 20 minutes using asexual reproduction. So it's really good for some organisms. Now, I did just make a note before coming on the live stream. For triple only, there's some examples you need to know, and that is these three. So these three examples of organisms can carry out both asexual and sexual reproduction. So you need to learn about those if you're doing biology only. So first of all, there's the protists, or parasites that cause malaria. Those are protists. I'm not sure actually if I wrote the right word there. I think it might be meant to say protists. I'll check. But anyway, some of them reproduce asexually when they're in humans, and then they produce sexually when they're in the mosquitoes. So they use both. Many fungi, remember that group of classification, they can reproduce asexually by spores, and spores are just those tiny little microscopic things that sort of bud off, and they're in the air. You can breathe them in and things, not very nice. But anyway, that's asexual reproduction. They can also sometimes do sexual reproduction, and that's to produce variation. Finally, some plants can reproduce asexually. For example, strawberry plants, they send out runners, and daffodils, they have bulb division. Bulbs are just the bits at the bottom. But they can also reproduce sexually and produce seeds from that. Okay, I think that's it. So Ellie, that was the last request. Okay. GM crops, I've done, and tissue culture, I've already done, so you can see lots of that. Oh, thank you so much, Majid. I really appreciate that, and thank you for watching the separate videos too. Yeah, they take a lot more work because I do the questions at the end, and I also do the little introduction and things with me standing there being all silly, but thank you, I really, really do appreciate it. Oh, thanks. Here's my rings. Getting sidetracked now. No, what about the endocrine system? Oh yes, Daniela, I'm sorry, I did forget that. Okay, I will take just 1 minute or 2 minutes to answer that. So the endocrine system, it comes up way at the start of homeostasis and response. Here it is, endocrine system. So the endocrine system is the second system of control in the body. The first was your nervous system. The endocrine system is based on glands and hormones, and then target organs. So glands are organs in the body that can produce hormones. Hormones are special proteins that can travel in the blood to a target organ, and they will cause the target organ to produce some sort of response. So your classic example is regulation of blood glucose levels. So for example, the gland would be the pancreas, and the hormone that it produces would be insulin. And if you're studying higher tier, you'll also learn that it produces glucagon. But let's stick with the insulin example because that applies to everybody. So the pancreas is the gland, it produces the hormone insulin. Insulin then travels in the bloodstream. When it gets to the liver and the muscles, it will cause both of those to take in the glucose and convert it into glycogen. it also causes other body cells to take in the glucose and use it in respiration. So those are one example of how the endocrine system works. There are others. We did briefly discuss adrenaline and thyroxine. That was way back at the start of the live stream. If you'd like to learn more about those, I didn't finish writing it down, but that's all about the fight and flight response. But anyway, flick back, and if you're doing higher tier, you can look at those. The last thing is that you need to know the placement of these 6 glands in the body. So you could be asked to identify those. So if you want to take a screenshot, you can. We've got the pituitary gland up beside the brain. The thyroid gland is in your throat. The pancreas sort of sits around the middle. The adrenal glands are above both of your kidneys. The testes in the male body are down there in the crotch. And the ovaries are in the female body, and that sort of sits over here. Okay, so that is everything really for the endocrine system. You could get a little comparison between the endocrine system and the nervous system. The endocrine system is a lot slower because you're relying on hormones traveling in the blood, which is slower than electrical impulses, which are ridiculously fast. But the good news about the endocrine system, sometimes the good news, is that the effects are longer lasting. So because you've actually got a chemical, like the hormone, working, it takes time for that to be broken down. Sometimes that's good because you get a longer lasting response, but sometimes maybe that hormone's hanging around and doing something whenever you don't need it anymore. But that's normally when negative feedback kicks in to bring things back to the set point. Okay, I think that's it, Daniela. Let me just flick through the questions for the last couple of minutes and then I'm done. Okay. Aisha, you want to know, where did you get your right hand ring from? I'm not sure which way round is right hand for you. So this one is from Pandora, I think about 2 years ago. And this one was a present. It's called a claddagh ring. I'm from Northern Ireland, and it's an Irish ring. It's you can see the sort of Celtic style details. It symbolizes loyalty from the crown, love for the heart, and I think friendship for the hands. And there's a lot of details about how you can wear it and what that means, but I just ignore those because it's a bit silly. But thank you very much. I'm glad you liked them. Sorry to everybody else that wasn't interested in that. Aisha, sorry if I said your name wrong. What's the difference between glycogen, I think you're trying to say, and glucagon? I'm going to write those words in the comments so you can see the correct spelling, because examiners will they want to know that you have exactly the right spelling. You won't get the mark if you spell it wrong. So glycogen is this way that humans store glucose. So we turn our glucose into glycogen to store it away for later. And that happens in the muscles and the liver. So that's glycogen. Glucagon is the hormone that causes glycogen to be turned back into glucose. My way of remembering the hormone glucagon and what it does is that it sounds like glucose gone. So when glucose is gone, you're going to need to get some more. So glucagon comes along and turns glycogen back into glucose. And that happens in the liver. So that's a little hint, might help you. You don't need to say sorry for getting it wrong. Everybody makes mistakes all the time. It's a good thing. It means you learn something. Okay, everybody, you can do it. Justin, you can do it. I have a lot of faith in all of you. The fact that you're here getting some help means that you care about your GCSE. So that's already so much more than lots of people. So don't stress it. Make sure that you have a nice bit of downtime before you go to bed so that you have a good night's sleep. All of that information really cements itself in your head and you wake up refreshed for your exam tomorrow. I have mentioned it before, but I've got a nice video that it's 1 hour 42. So if you really, really need some extra help, yeah, if you started it now, you might finish it at a decent time. I don't know, speed it up and you'll squish it down. But thank you so, so much for watching. Please subscribe. It really, really helps me get my videos out there. I know they're not going to be useful for you soon, but they will help next year's students. So it will mean a lot to me if I got some more subscribers. I don't have a lot yet. But thank you so much for watching, and massive good luck. You can all do it. Okay, thank you everybody. Bye.